3, 2.
Yeah, we're alive.
Good morning, everyone. Welcome to the Clemson University Graduate Research and Discovery Symposium final competition on April 16th, 2021. I'll be your host today. I'm Nick Gregorich, and I'm the Director of Research Initiatives for Graduate Student Government here at Clemson University. Hello to all those in Twitch world and also in Zoom world. Thanks for being here. It's 9 a.m. and it's time to get this competition rolling. The last 13 months have undoubtedly been strange. And although the pandemic has unfortunately turned this into a virtual competition and affected us in so many different ways, I and many others are very proud of how this has come together. Back in January, we were struggling to come up with a format for this competition that would incorporate both posters and oral presentations and make sense in a virtual setting. This was the first time doing grads in a virtual space, and we truly had no idea how we were going to do this. In comes Jan Lay, who passes along the idea of incorporating these personalized Google sites on the internet. These personalized Google sites allowed students to upload a 2-minute video, a poster that would otherwise be printed off, an abstract, a photo of themselves, and information about their project, their advisor, their department, and the college that they fall under. We then took these Google sites and incorporated them onto our GSG GRADS website that looked really professional and had a bunch of thumbnails and divided by all of the colleges to display all of the posters for the competition this year. It was a fantastic way of displaying all of the fantastic research that all of our grad students are doing across this campus, across all disciplines of study. After the preliminary phase of this competition, we had 30 contestants from all 7 colleges. 2 contestants from each college advanced to the final round of the competition today, and we are now down to 14 finalists. So what is the format for today's competition? Today's competition is 7-minute research presentations that are geared towards a non-specialist audience. Grads would normally be done in the Watt Auditorium with posters, presentations, and be an all-day event. We still think that this way of hosting grads is a fantastic way of showcasing the wonderful research conducted by our graduate students here on campus. So what are the awards for today? For the first time, the GRADS champion will earn the $1,000 grand prize. That's right, you can earn a grand with GRADS, as my committee would say. Second place will earn $500. Third place will earn $250. And this year, we've incorporated something new called the People's Choice Award, where the audience will be involved and have the opportunity to text in their favorite presentation at the end of this competition, which will be around 12:15. We hope you are there to participate in this and vote for your favorite presenter. A note to the contestants. All awards from the prelims and finals will be handed out in the coming weeks, so be on the lookout for an email from me. Putting on an event like this is no small task, and it took a lot of moving parts and a lot of fantastic people. Although I get to sit here and host you guys for the next 3 hours, I would not have been able to pull this off without all the fantastic people on this slide. I want to take a second to thank the GSG Research Initiatives Committee. My assistant deputy, Eric Antonio, has been a huge asset to the committee for the past year. Having been on this committee for over a year, he was a huge asset, a great partner, and an overall huge contribution to making this competition happen. Thank you, Claire Texter. Claire took control of the survey forms for both the preliminary and final phases of this competition. If you've seen as a judge, they were done in Qualtrics and they look very professional, they're flawless, and they're very crisp. Quite honestly, if Claire wasn't on this committee, I have no idea how we would have made surveys for this competition, because typically we would be printing these off on paper. Thank you, Claire. I want to thank Vishal Thomas. Vishal set up our Canvas page for the competition this year, which served as a huge tool for our competitors. This allowed us to store important documents, links, information, and anything else we needed to get to our contestants to let them know what was going on with the competition. Thank you, Saida. For those in Twitch world, we have this really swanky overlay this year with a Clemson paw. I'm looking at it right now, an orange box, and our new GRADS logo on the bottom. Quite honestly, it looks like you right now, and I think it looks really cool. Thank you so much, Saida, for taking that on. Thank you, Simeon Babatunde. Simeon was in charge of the People's Choice Award this year. I wasn't really sure what to expect with this, but Simeon found a fantastic software that was affordable and suited this competition and will be great to add for future competitions that we have with GSG. Thank you, AP, for staying on as the prior director and for providing information that I hadn't known if I had otherwise not been on this committee before. I also want to thank Libby and Pinar for attending all meetings, contributing when needed, and always being great members of the committee. I also want to thank John Porter, our vice president of GSG. John Porter, who is our resident DJ of virtual GSG events. He'll be keeping the crowd noise under control, spinning music on the beats on the ones and twos. And he has also been a great insight at all of our meetings. Thank you, John. I want to give a huge shout out to Amal Panathil. Amal was the mastermind behind building our website that was used for the preliminary phase of this competition. It looks absolutely fantastic. And quite honestly, you could have convinced me that we hired someone to do this for us. Thank you, Amal. I want to thank Dean Lopes. Thank you for taking time out of your day today to be here today to provide the opening remarks for this competition and being so supportive for all phases of both this competition and the 3-minute thesis competition. I can't wait to continue this relationship as we move into the summer and plan for 3MT in the fall. I want to thank all of those that were part of the Grad 360 rehearsal sessions that we had leading up to the final competition. I have no doubt that these sessions adequately prepared our competitors for the competition and provided them great advice and skills to prepare them for today and becoming a better presenter. For that, I want to thank Dr. Lori Pindar, Dr. Cameron Bushnell, and Jan Lay. And again, one more shout out to Jan Lay for creating the preliminary format for this competition. Thank you so much, Jan. I also want to thank Jill Bunch for again promoting and advertising this competition. She did this for both 3MT and GRADS, and I thought she did a fantastic job. I also want to thank Ellen Graben, who I believe was the designer of our brand new GRADS logo for this year. And I also want to thank Rebecca Tao. There are two people in particular that I want to highlight in the planning of this competition. They've been in the trenches with me since day one, and I'm greatly appreciative for the amount of time that they have spent with me in making these competitions as great as they've been. Thank you, Jody Cox from Grad 360. Your support, guidance, and recommendations and attention to detail are greatly appreciated. Your devotion and commitment to professional development of graduate students at Clemson University is exceptional. Thank you so much for putting on the rehearsals and planning those for our contestants. I heard that 9 out of 14 people came through, and I have no doubt that those 9 people are adequately prepared and will do fantastic today with this competition. It has been a pleasure working alongside you and learning from your experiences, and I can't wait to continue this with 3MT in the fall. Thank you so much. Last but not least, I want to take a second to thank Dr. Tia DeMoss. The guidance, support, and mentorship you have provided me in the year and a half that I've known you is absolutely phenomenal. Thank you so much for taking time out of your week to mentor me in this role and mold me into a better leader, and most importantly, a better person. Your fingerprints are again all over the planning of this competition. We are so appreciative of the time that you take out of your day to make these as great as they have been. I'm so excited to continue learning from you as I enter my last year as a Clemson graduate student, and thank you so much. With that being said, we have our very special guest today that will be leading off the opening remarks of this competition. And with that being said, Dean Lopes of the Graduate School, thank you for being here, and the floor is yours.
Thank you, Nick. I appreciate this opportunity. Can you all hear me okay? Am I coming across all right? All right.
Yes.
Thank you. So I just want to start off by acknowledging that these are fantastic events, and I certainly look forward to them, as I think all of us do. But it's important to recognize that we do live in a very complex world, and what we do at work is only part of our everyday lives. And so there's a lot of good that happens, but there's also a lot of pain that's happening. And I think we need to recognize that and understand that we are here to lean on each other for support. One of the things that I've been most impressed with since joining the Clemson community is just how much students take this to heart and really serve as support for each other. It's heartwarming, and it means a great deal. So let me move on to the agenda. I want to echo everything that Nick said in terms of acknowledging all the folks that came together to make this happen. I think any enterprise this complex takes a great deal of teamwork. And so I hope you heard in his words what I view it as highlights, the concept of teamwork, the concept of innovation and adaptability. You know, we've had to change directions constantly throughout this past year and be really creative in terms of how we continue to do the things that we do in the best possible way. And I think this event is really a manifestation of all of those things brought together to put on a fantastic event. I thank Nick and Eric Antonio for and their wonderful committee for all their hard work in bringing this together. I also want to highlight the GRADS prep is really supported by a lot of individuals who do a tremendous amount to help students in their research, faculty, mentors, associate deans at the colleges and the grad school, and the deans. Nick at one point reached out to me and asked, to sort of help folks remember that we were putting this on. I reached out to the deans and they immediately marshaled getting information out to students to make sure that everybody had an opportunity to compete today. Also want to acknowledge Drs. Cameron Bushnell and Dr. Lori Pindar. I think we recognize these individuals as really extraordinary and accomplished, dedicated scholars who are at the top of their disciplines. They're definitely beacons of excellence and pride for Clemson. But I think the one thing I've come to recognize of these two individuals is that they show us that you not only set high standards for what we do, but you can also be compassionate. It's not a binary decision. I think we can do both, and they exemplify that. Really grateful for the technical expertise of Jan Lay. I don't know that we've met. We may have met over Zoom. I've met about 5,000 people over Zoom since I moved to Clemson. So I may have interacted with Jan in some capacity, but I look forward to interacting moving forward. And I want to highlight, so every day I get to come to work and work with fantastic staff at the graduate school. And I got to tell you, I never take for granted the fact that I get to work with phenomenal individuals. So, Jody Cox, Rebecca Tao, Ellen Graven, Jill Bunch, all mentioned, and of course, Dr. Tia Dumas. These are extraordinary individuals who care greatly about our student success. And I can promise you, I know it's their first thought when they wake up, it's their last thought when they go to sleep. And I'm really proud to work with them. So let's talk a little bit about GRADS. I think there's several elements of this that appeal to me, and I hope will appeal to you throughout the day. The one thing that sort of comes to mind is that this is a highly selective, highly competitive process. And sometimes we kind of forget about the fact that we do live in this very competitive environment. I think all of us are here precisely for that reason. I competed in these types of events as a graduate student. I think many of you probably know that I attended another great institution in the state of South Carolina, but I did actually come up to Clemson University for some statewide competitions that focused on the research that we do, and I won a couple of those competitions. But in general, I think we always want to compete. I mean, it's kind of what we do, right? When we publish papers or when we put out our scholarly works, typically, if what we're doing is really good, and it is, there's always somebody else who we're competing with who's trying to get it out, faster than we are. So I've always kind of enjoyed that whole concept of scooping and always tried to be on the scooping end of it instead of the being scooped side of the equation. But I think that's part of it. And I think that kind of competition is what spurs us to be better at what we do. And so I love the fact that that's a component of this. But we shouldn't lose sight of the fact that just by competing, you're already winning, right? Because you're showcasing the amazing work that goes on this campus, the amazing work that you do. And by its very nature, that means that you're all in. So let me tell you why this is important, if you don't already know. We're very proud of the fact that Clemson University is an R1 research institution, right? That puts us in a whole different league and with the most elite universities around the country. The reason we're there is because of the graduate students. The work that you do and the quality of work you put out and the recognition that it gets is all part of the equation that goes into deciding which universities are R1 status. And so we know, and the campus recognizes, the university leaders all know this, that graduate education is an integral part of what makes Clemson extraordinary. You not only contribute to the knowledge that our world has access to through the works that you do, but you also contribute to the educational excellence of Clemson University. By virtue of the fact that you're in classrooms, in labs, and studios, and you're part of educating students. You also lead all of the creative inquiry activities that go on our campus. So graduate students are complex, and they work hard, and they bring a lot of recognition to the campus. And I can promise you that not a day goes by that we don't all recognize that. The other part of this that's really important is that you're learning, and this is a lifelong learning process, I promise you, because I still do it. You're learning how to both showcase and describe the very complex work that you do to a general audience. This is really, really hard, and it's really, really important. I remember that as a graduate student, my parents would ask me what I would work on, and I'd show them x-rays, and I'd show them graphs, and I'd show them papers I published with words that they didn't understand. And we always tried to impress folks largely through the complexity of the things that we do. That is the wrong approach to take. That's what approach we take internally within our own disciplines. But to make our work mean something and to make it approachable, we have to be able to communicate it to much broader audiences. And the folks who are the best at this are the most successful. And I believe that you all will be very successful at this moving forward. So I want to finish on that note. I'm looking forward to hearing the presentations today. And I just want to echo that we are also very, very proud of you. So thank you all. Nick, it's back to you, buddy.
Thank you again, Dean Lopes, for being here today. That was really nice. Okay, it is time to get this going. So Tristan Schramer, why don't you go ahead and share your screen? I will stop sharing mine. Hold on.
Sorry, I had to unmute myself there. Okay. Can everybody see that all right?
Okay. So first up, we have Tristan Schramer. Tristan is a master's student in biological sciences in the College of Science. His faculty advisor is Dr. Christopher Parkinson. And today he's going to be talking about venom profiling of a rear-fanged snake, Canophis lineatus. Can I get a
Thank you.
check? Can I get a mic check, Tristan?
Yeah, can you hear me all right?
Yeah. Okay. Whenever you are ready to begin, please feel free to do so. And good luck.
Hey, everyone. As was just said, my name is Tristan Schramer. I'm going to talk about profiling the venom of a Middle American rear-fanged snake and discuss how that led to the discovery of new venom proteins. Reptile venoms are complex protein cocktails that have diverse functions and biological effects. Learning about a venom's composition not only helps us improve available treatments for snakebite, but it is also the first step in unlocking venom's pharmaceutical and therapeutic potential. For instance, snake venoms are used or are being investigated for the treatment of a number of conditions, from reducing high blood pressure to stopping bleeding at surgical sites, among others. I've listed a few FDA-approved drugs derived from venom on this slide as well. Now, I'm sure we're all familiar with the dangerous front-fanged snakes, such as vipers, rattlesnakes, and coral snakes. But you may be less familiar with rear-fanged snakes, which are generally considered harmless in the sense that their bites are not fatal to humans. However, our categorization of snake species into deadly or not has hampered much progress in the study of venom and its evolution. We now know that venom is a much more broadly possessed trait within snakes, even among those traditionally considered harmless, and new omics technologies can help us investigate their venoms for the very first time. Here, we sought to investigate the venom of Canophis lineatus, a representative rear-fanged snake from Middle America considered mildly venomous due to its potent bite. However, its venom has never been studied. There are a few cases of envenomation in the primary literature, but the secondary literature is rich with anecdotal reports. The most commonly reported effects include an immediate burning pain, prolonged bleeding or hemorrhage for up to several hours, and substantial localized swelling, edema, and or inflammation that may extend the entire bitten extremity and last for up to 11 days, but usually just 1 to 3. Aching, numbness, stiffness, discoloration, tingling at the top of the head, pain in the axillary lymph nodes have also been reported. So obviously the snake could hardly be considered harmless. So we are interested in using transcriptomics and proteomics to explore its venom for the first time. So we first collected a single specimen from the Yucatan Peninsula in Mexico. Then we extracted its venom, shown here with capillary tubes over its rear fangs. And a few days later, we dissected its large venom glands highlighted in the CT scan here, so we could sequence what genes were being transcribed. Next, we combined RNA and protein sequencing to analyze the composition and expression of the snake's venom and explore the evolution of particular genes. Our final venom gland transcriptome showed that over 60% of the genes expressed in the venom gland were attributable to toxins, as expected. So let's break those toxins down. Now on this slide, don't worry too much about the acronyms. They just represent venom protein families. What really matters here is the type of biological activity these toxins induce. Interestingly, in blue, we see that 55% of the toxins expressed in the venom gland were snake venom matrix metalloproteases, a just recently discovered venom protein family. Preliminary investigations suggest that they act as tissue degradation agents. And similarly, the second and third most highly expressed toxin families act as anticoagulants and cause tissue damage, respectively. Thus, the venom is predominantly what we call hemorrhagic, with over 85% of the venom components acting essentially as a chemical meat tenderizer. So it's no surprise that profuse bleeding and substantial localized swelling are associated with the snake's bite. Now, some of these pie slices are even composed of multiple genes, and we are able to look at the expression of each gene independently. So in this pie chart here, or in this chart here, each bar represents a single gene and its expression. In addition, an asterisk above a bar indicates that we were able to validate the presence of this toxin in the venom using proteomics. What is most interesting here is that 3 of the 5 matrix metalloproteases in blue represent an entirely new variant, including the most highly expressed toxin in the venom. Previous investigations discovered 2 types of snake venom matrix metalloproteases, colored in red and orange, which evolved independently in 2 different groups of rear-fang snakes. When we re-examined the evolution of snake venom matrix metalloproteases, including the new variant, colored in yellow, we discovered a 3rd independent origin in Canophis lineatus. This novel variant differs from the previously described forms by having a unique partial gene deletion. This evolutionary novelty likely leads to similar functional novelty, including distinct pharmacological properties. Rear-fang snakes make up over 2/3 of the world's living snake species, but only a small proportion of venom studies. Right here in our own backyard, the hemorrhagic venom of the pygmy rattlesnake, similar to that of Canophis lineatus, is used to treat heart attacks by preventing blood clots. So it's easy to see how there is tremendous opportunity in studying the venom of rear-fang snakes, both evolutionarily and as a treasure trove of novel bioactive proteins and variants that may have unique pharmacological uses. Our study underpins how even a single specimen can be used to discover these unique toxins, and additional sampling will only unearth even more variation. And with that, I'm happy to take any questions. Thank you.
Thank you, Tristan. Great talk. So we have a question from the committee here. As you presented incentives to research the venom of harmless snakes, what features could make researching a particular species of harmless snake a priority over other species?
Yeah, that's a good question. So the interesting thing about harmless snakes is that their venom delivery system is a little bit different than that of the front fang snakes, which can store larger amounts of venom and deliver it essentially as a bolus of venom all at once. Where with rear fang snakes, they often have to chew their prey for a bit of time to deliver that venom. So because they can't deliver as much venom, they essentially have often developed even more potent venoms per volume. And these venoms are often extremely specialized on the prey they eat. Not only that, but their venoms are a little more simple than those of the front fang snakes. So it's a little bit more of a tractable system in understanding, the evolution of certain genes, but also linking function to specific components of the venom.
All right. Thank you, Tristan. Great job.
Thank you.
Okay. With that being said, we will now give our judges another minute to go ahead and finalize their scores. And next up, we have Pawanjit. Pawanjit, if you could please go ahead and share your screen, and we will begin in a moment.
Can you see my screen?
Yep. So next up, we have Pawanjit Sanhu. Pawanjit is a master's student under the advisement of Dr. Nishanth Thorayal in the Department of Plant and Environmental Sciences, and she is in the College of Agriculture, Forestry, and Life Sciences. And today, she's going to be talking to us about inducibility and specificity of glyphosate resistance in Palmer amaranth as elucidated by global metabolomics approaches.
Testing, Pawanjit.
Sounds great, Pawanjit. Okay, whenever you are ready, please feel free to do so, and good luck.
Thank you. So first of all, what is glyphosate? So glyphosate is a herbicide that is the active ingredient of the popular trademark Roundup. So this is the most commonly herbicide that we use in our lawns to kill the weeds. So not only our lawns, but agriculture around the world is also heavily dependent upon glyphosate, as it is the most widely used herbicide in the world. For example, in United States, more than 90% of area under corn, soybean, and cotton, which collectively contributes around $10 billion every year to US economy, is under herbicide-resistant cultivars, most of which is managed by glyphosate. However, many weeds around the world have developed resistance to this herbicide. That is, the weeds are no longer killed by glyphosate. So as you can see in this map, glyphosate resistance has occurred across 6 continents in the world. Further, within the United States, it's almost everywhere. So this is a major threat to our agriculture and food production systems. So there is a need for knowledge-based decision-making to devise weed management strategies, for which we need to understand the herbicide resistance. So that is where my research comes into play. I try to understand the physiological differences between resistant and susceptible plants. That is, how the functioning of resistant plants differs from that of susceptible plants at a cellular level. So some of the specific questions that I am interested in are, is the resistance mechanism always active or activated in response to herbicide? That is, whether the switch of resistance mechanism always on versus that it is off but turned on by herbicide. So this has energy concerns similar to electricity. That is, if the resistance is always on, it would have more energy cost to plants compared to that if it is turned on by herbicide. However, here this could be more useful to us because we are trying to kill these plants. The other question that I am interested in is, does resistance to glyphosate help plants cope with other stressors? That is, the resistant plants that can survive glyphosate, can they also survive other stresses in the field, for example, drought or nutrient stress better? If so, this would turn these resistant plants into what I call the super weeds, because now they have increased general immunity against stressors, which poses further threat. So the answers to these questions are important in knowledge-based decision-making for devising weed management strategies. So now, how do I answer these questions? So to answer these questions, I use global metabolomics approaches to capture the cellular physiology and functioning of plants. So these global metabolomics approaches act like a camera which tries to take a snapshot of the abundance of all the metabolites in the plants. So metabolites are basically all the small molecules that are involved in plant metabolism. For example, sugars, organic acids, amino acids, secondary metabolites, etc. So these secondary metabolites are the pigments that we see around us as colors in like fruits or vegetables, basically every color in the nature. So many of these secondary metabolites act as antioxidants. So these are the same antioxidants that are recommended to us by doctors for our good health. So these are actually produced in plants to provide plants the good health, that is the capacity to tolerate stress. So in general, higher the abundance of antioxidant means higher the capacity to tolerate stress. So to conduct this study, I took multiple susceptible and resistant plants of Palmer amaranth, which is my study species, and exposed them to 3 different treatments: the glyphosate treatment, another set of plants to drought treatment, which served as general stress and helped me answer my second question, and then third set of plants to no stress or control treatment. From all the 6 treatments, I captured the global metabolite profiles. So now moving on to the results, the first question that I was interested in is the resistance mechanism always active? That is, the switch is always on versus that it is turned on by herbicide. So to answer this question, I looked at the abundance of a metabolic marker of glyphosate stress. So this metabolite actually accumulates when the plants are experiencing stress from glyphosate. That means higher the accumulation of this metabolite, higher the stress from glyphosate. So in this graph, the y-axis indicates abundance of that metabolite and the x-axis indicates different treatments. And the gray bar represents control treatment and the red bar represents glyphosate treatment. And the first 2 graphs are for resistant biotype and the next 2 graphs are for susceptible biotypes. So as you can see that in the resistant plants sprayed with glyphosate, there is increase in abundance of that metabolite indicative of glyphosate stress. Although this accumulation is lower compared to what was observed in susceptible plants, which eventually die from glyphosate, nevertheless, this means that resistant mechanism was not activated at the time of spray of glyphosate and is activated after exposure to herbicide. Now, moving on to my next question, does resistance to glyphosate help plants cope with other stressors? So the answer to this question is both yes and no. So I'll explain how. So to answer this question, I compared the abundance of secondary metabolites that act as antioxidants, as I mentioned before, across different treatments in resistant plants. So as you can see in this graph, the x-axis represents the total abundance of these metabolites. So as you can see that in control and drought treatment, the abundance of these metabolites is not different in the resistant plants. So this means the question that whether resistant plants that can survive glyphosate can also survive other stresses, the drought better, is no. However, in the glyphosate treatment, that is after the plants have been sprayed with glyphosate, there is a humongous increase in the abundance of those metabolites. This means that the resistant plants that have already survived glyphosate might survive those stresses better, which is another question for me to test. So to sum up, the glyphosate resistance mechanism is activated after exposure to glyphosate, that is, it is inducible, as mentioned in my title slide. And the glyphosate stress specifically and not drought results in accumulation of secondary metabolites that might provide higher general stress tolerance. Thank you, and I will take any questions.
Thank you, Puanjit. Great talk. So, question from the peanut gallery. Why do you think the glyphosate-treated R biotypes have a richer phenol propanoid profile than the S biotypes?
So yeah, so the phenyl propanoids are the antioxidants that I just presented. So because one of the reasons that could be because the resistant plants, they have higher copy numbers of the enzyme that regulate those phenyl propanoid pathways. That is actually the mechanism of glyphosate resistance in those plants. So that could be the one of the reasons that they have the higher abundance of those enzymes, higher abundance of those metabolites, because they have higher abundance of the enzymes that are regulating those pathways.
Awesome. Thank you, Puanjit. So we will now move on to our next competitor, who is Paritra Mandel. And in the meantime, we will give our judges another moment to go ahead and finalize their scores before we move on to the next presentation. Go ahead and share your screen, Paritra. All right. So next up, we have Paritra Mandal. Paritra is a PhD candidate under the advisement of Dr. Terry Bruce, and she's in the School of Computing, Biomedical Data Science Informatics, and she's in the College of Engineering, Computing, Applied Sciences. And today she's going to be talking about robust exosomal biomarker panel discovery in ovarian cancer using machine learning approaches in studying miRNA-mRNA target interactions.
Thank you, Nick.
We could get a mic check from you, Paritra?
Yes, please. It's Paritra testing.
All right. Thank you, Paritra. Whenever you are ready to begin, please feel free to do so, and good luck.
Thank you. This presentation is directly speaking to women and then their loved ones, which really means it's speaking to all of you. Today, I'll be talking about the perils of ovarian cancer and how to protect our women by creating awareness about its risk and the importance of early detection. So don't get swayed by the technicality of its terms in the title just yet. Now, ovarian cancer is lethal, being the 5th leading cause of cancer-related deaths among women, according to the American Cancer Society. With 22,000 new cases being diagnosed each year, it is the 2nd most gynecological cancer in the United States. The 5-year survival rate is at a 48.6%, meaning of all of the people that are diagnosed with ovarian cancer, less than half of them have a chance at surviving this disease at 5 years. Now, if it's diagnosed early, this 5-year survival rate is increased to a 93%. So this makes the case that early detection is extremely important in ovarian cancer. And at the same time, it is extremely difficult to do so because of its asymptomatic nature of the disease. And most cases are diagnosed when the cancer is already metastasized into later stages. So let's talk about treatment. So treatments in ovarian cancer are mostly invasive in nature, and you would want your treatment to be as non-invasive as possible. So how do we do that? We do that through liquid biopsies. Now, liquid biopsy is examining body fluids. So, body fluids are excellent sources of information, and they can be used as effective early biomarkers. Now, biomarkers, you're going to hear that a lot. What is a biomarker? So think of it as like a fingerprint of a disease state or a process. And these could be genes, proteins, or miRNAs or small RNAs. Now, for example, if I say gene A is causing a certain disease, this means that gene A is a biomarker for that disease. Now, the next slide shows you the different approaches that you can do for biomarker discovery. So there's genomics, proteomics, lipidomics, and depending upon what it is that you're looking for. So for example, existing biomarkers in ovarian cancer are the CA125, but that has a low predictive accuracy. So the image on the right shows you the different types of biopsy, liquid biopsies that you can use for examination. Now in this slide, I want to give you a bird's eye view of my research. So on the left, you see these ovarian cancer cells and that they contain certain proteins and RNAs, and they're kind of like carried off by these taxis called exosomes, and then they transfer that information to normal cells. And as you can see, these are slowly turning into cancerous cells. Now, what we did was we collected these body fluids from the cancer and the healthy individuals, and then we extracted their exosomes right here. And then that resulted in a data matrix such as this, which then helps us to apply data science to it to find the most informative markers. This slide shows the different subtypes of ovarian cancer and makes the case for the heterogeneity of the disease, which means that it's difficult to diagnose and it's also difficult to treat. So to bypass this complexity, there is a growing need, growing interest in cell-derived particles that are participating in intercellular communication. This brings us to the start of the show, which is basically introduction to exosomes. Now, these exosomes are so important, they're so interesting as they carry different kind of messages to the neighboring cells. So as you can see, they're pinched off from the cell. They were the taxis that I was talking about in the previous slide, but just that they're not taxi-like, they're membrane bound instead and carry the same surface membrane proteins as the parent cell. So they are very important and very interesting because they carry genetic information and transcriptomic information from one cell to the other. So what did we really do for this study? We collected 42 cervical mucus from 42 individuals. We then extracted the exosomes from the cervical mucus using a novel method that my lab developed using capillary channel polymer fibers. And then once we collected them, we sent them for small RNA sequencing to see what sort of small RNAs are in them, which resulted in a data matrix such as that. And then that enabled us to apply various supervised and unsupervised machine learning approaches to find out, identify the best features. Now, each method gave us a group of potential candidates. And what we were interested in is to look at if there are any common markers that each method gave us. So indeed, there were 4 markers that lay at the intersection of those methods, and we used them to generate our biomarker panel. Now, this is our biomarker panel consisting of 9 biomarkers, 4 of which we just talked about, and 5 of them were differentially expressed genes differentially expressed mirs that differentiated between tumor and benign samples. So what did we do next? We wanted to test the ability of this panel on an independent validation cohort, which was tissue specific, to see if they're able to differentiate tumor from benign samples. And we did get a really good classification accuracy. Now, in nature, data science is not enough, and we need to functionally and biologically validate this sort of a panel. And so therefore, we looked at the causal relationships of these miRNAs directly with the disease, and we found 2 of them that were causal, and the rest of them that were involved in some sort of ovarian cancer pathogenesis or tumorigenesis. And we also did pathway enrichment analysis, which led us to successfully functionally validate our panel. So as a summary, what we did was we identified an exosomal biomarker using our lab data. We validated that on an independent data set, and we also validated that biologically. And as a final application of my research, what we want to really do is come up with a screening platform, which is much like a pregnancy test that would capture specific marker proteins on the surface of the exosome. So I just want to highlight a couple of key points of my research, and that our method is non-invasive and inexpensive. We're using exosomes to perform early detection of ovarian cancer and eventually other types of cancers. We're looking at the miRNAs. We're looking at the cancer from an RNA level, non-coding RNA level. At the same time, harnessing computer science and data science to resolve biomedical problems to save lives. So, and we know that cancer is smart. So we need to, in order to defeat it, we need to be one step ahead and be really aware. And as I leave this presentation, I want you to keep in mind that the ribbon color for ovarian cancer is teal. Thank you.
Thank you, Paritra. Great talk. Okay, question from the crowd. What might it take to scale up this test so that it can be publicly used? You're muted, Paritra.
Sorry, I was muted.
That's
So
okay.
yes, great question. And that's one of the it's one of the problems that we go through because the sample sizes are really small. So what I'm doing with this current research is looking at different data sets, these, collection of these small data sets that give us some information, and kind of working on an aggregation protocol that sort of gives a global rank to this, to these biomarkers throughout different studies and across different platforms. And that's a question that I'm trying to answer at this moment. So I hope that answers the question in some way.
Great. Yeah. Thank you, Paritra.
Thank you.
Great job.
I got the keys as well.
Okay.
See ya.
Okay. With that being said, thank you, Paritra. And we will give our judges another minute to go ahead and wrap up their scores. And then we will move on to our next competitor, Sarah Krivasic. If you want to go ahead and share your screen, Sarah. Shout out to DJ Jump Order on the ones and twos, spinning the smooth grooves in between competitors. Good stuff. Okay, so next up we have Sarah Kravasik. Sarah is a PhD candidate under the advisement of Dr. Kristen Scott and Dr. Thomas Zaganchek, and she's in the Department of Management and Organizational Behavior in the College of Business. And today she's going to be talking to us about self and other compassion, moderating effects of emotional exhaustion and perceived receipt of help on interpersonal citizenship behavior.
This is Sarah testing.
All right, sounds great, Sarah. Whenever you are ready to begin, please feel free to do so, and good luck.
Thanks again. This is Sarah Kravasik, and I will be presenting my research that I'm working on with Dr. Kristen Scott and Dr. Tom Zaganchek. So first, just starting at a high level, what is compassion? This is a term that most of society is familiar with, but might be out of touch with. And the need for compassion is more important today, now more than ever, while we're facing this pandemic. The need for compassion is important for medical professionals who are risking their lives every day. The need for compassion was important, although not necessarily put at the forefront at our most recent political election. And the need for compassion is important when facing social justice issues revolving around race. So there's this growing need for compassion, both for ourselves and for others. So how does research conceptualize compassion? Well, it's separated into two components, self and other compassion. And self-compassion is defined as kind acts towards the self. And an orientation and focus for someone who scores high in self-compassion will be around the self. And an example of this might be forgiving yourself when you make a mistake at work. In contrast, other compassion refers to kind acts towards others. So the main difference here is that there's this orientation and focus around others. And an example might be empathizing with someone struggling at work, so you try and help alleviate their suffering in some type of way. So while these two components conceptually differ, one thing that they have in common is that they're comprised of three main components: kindness, common humanity, and mindfulness. Kindness refers to either self-kindness or kindness in the form of others. Common in humanity is the idea that we all experience and are in this shared experience. So we all make mistakes as individuals, we all fail, and also suffering is part of the larger human experience. And finally, mindfulness is the balanced awareness of the present moment. So delving a little bit deeper into our research question, we are interested in compassion and how it's related to workplace outcomes. So our first research question, we asked, will employees high in self or other compassion engage in helping behaviors in the workplace? And we refer to helping behaviors as citizenship behaviors in our research, but basically this is the idea that an employee will engage in behavior that is outside their immediate job duty. So an example of this might be an employee stays late after work to help another coworker catch up with his or her work. So after examining if these relationships exist, we went a step further and said, okay, is there a limit to the amount of helping an employee with either of these traits will engage in the workplace? And we examined two moderators. First is emotional exhaustion, which is one component of burnout. And second is perceived receipt of help. And this is the degree to which we perceive another individual as helpful or not in the workplace. So just briefly touching upon the theoretical framework, we draw from conservation of resources, or core theory, and this posits that individuals are motivated to gain, maintain, or attain resources in order to avoid depletion, because when depletion of resources happens, then stress ensues. And there's two important principles to core theory that I wanted to call out that will help connect the dots of why we chose these moderators. So the first is the desperation principle, and this is the idea that individuals will make irrational decisions whenever they're depleted of resources and they may self-preserve. So we chose emotional exhaustion because we are interested if these individuals high in these traits are experiencing high levels of exhaustion, will they not engage in these helping behaviors? Another important principle to core theory is the resource investment principle. And this is the idea that employees will invest resources in order to gain, maintain, or attain resources later on in the future. So we chose perceived receipt of help because we had the thought and question, if employee invests resources in the form of helping behaviors now, do they have the perception that another employee will help them later on down the road or do them a favor in the future? So now going on to the findings, I'll first start with self-compassion and that first moderator, emotional exhaustion. So basically what we're seeing here, so whenever we're experiencing low levels of this burnout component of emotional exhaustion denoted by the dotted blue line, we're seeing that helping behaviors are going up, particularly for those who score high in self-compassion. And in contrast, whenever we're experiencing low levels of emotional exhaustion, which this is actually denoted by the dotted blue line, the high levels Oh, actually, let me reverse. When we're experiencing high levels of emotional exhaustion, which is the solid line, we're seeing that helping behaviors are actually decreasing, particularly for those who score high in self-compassion. Moving to the second moderator, perceived receipt of help. So whenever we have a high perceived receipt of help, so we perceive that other individuals are helpful towards us in the workplace, we're seeing that citizenship behavior or helping behavior is going up, particularly for individuals who score high in self-compassion. But in contrast, whenever perceived receipt of help is low, so that we do not perceive other individuals as helpful towards us, we see that helping behaviors goes down, particularly for those who score high in self-compassion. Two important findings I wanted to call out for other compassion. So we found a positive and significant relationship between other compassion and helping behaviors. So what this means is as an individual scores higher in other compassion, they're more likely to engage in helping behaviors in the workplace. The second important finding, which was not in line with our hypotheses, was that there was no significant interaction effect among the predictors. And what does this mean? This means that regardless of how emotionally exhausted an individual is or the degree to which they perceive another individual as helpful, they're still engaging in these helping behaviors in the workplace, even if they are exhausted or don't have a high level of perceived receipt of help, if they score high in other compassion. So tying this all together, why is this important? Well, first, for self-compassion, research says that we don't self-prioritize. There's these conceptual arguments that saying there's not a trade-off that exists between helping ourselves versus helping others. And our empirical results are conflicting with these conceptual arguments as we're showing, well, we might really only help others when we're not emotionally exhausted or perceive others to help. And this has important practical implications. If organizations want to increase helping behaviors, they might implement interventions that are geared around other compassion. And this leads to the importance of these findings. So although the interaction effects were not in line with our hypotheses, these were super interesting to us as this really attests to the resiliency of this construct. And this is saying we help others regardless of our exhaustion or the degree to which we perceive others as helpful towards us in the workplace. And again, important practical implications. If an organization such as the medical profession wants to increase helping behaviors, then they might implement interventions geared towards other compassion. This leads to the conclusion of my presentation. Thank you, and may you go in kindness.
Thank you, Sarah. Great job. So we have our question from the crowd here. How would you reconcile your second takeaway, i.e., other compassionate will always engage in ICB, with the conservation of resources theory?
Yes, so that's a really good question. So we're seeing maybe for self-compassionate individuals that this depletion of resources is happening whenever they're engaging in these helping behaviors. But for other compassionate individuals, it's not so much of a depleting process to help other individuals. And it might even be stimulating or motivating or regenerative. It's kind of the notion of you do good, you feel good. So we might see that this depletion of resources isn't really occurring for these individuals who score high in other compassion.
All right. Thank you, Sarah. So with that being said, we are going to enter our first intermission here. And we ran a little over on time on the first session, so I'm not entirely sure if we're going to start right back up at 10:13, but I will keep everyone posted, and we will see you on the flip side.
Where we go.
Let everyone know that we are going to go ahead and start right on time. It is 10:13. So Tannen, if you could please go ahead and share your screen, and I will stop sharing mine. Before we begin, I want to let the judges know, thank you so much for being here. We are now done with the first session. So for those that are scoring the second session, you are going to want to go back to Eric Antonio's email and use the second Qualtrics link to do this session. And again, thank you so much for taking time out of your day to do this for us today. So next up, we have Taneen Haidari. Taneen is a PhD candidate under the advisement of Dr. Dennis Bosman in the Department of PDBE/CSM in the College of Architecture, Arts, and Humanities. And today, he's going to be talking to us about streamlining transportation project development process, PDP.
This is Taneen testing.
Thank you, Taneen. Sounds great. So whenever you are ready to begin, please feel free to do so. And as always, good luck.
Thank you, Nicholas. Transportation systems such as highways, roadways, bridges, intersections, even sidewalks are the arteries of cities and societies. It links you to your job, to grocery stores, to a market. It links you, your family, friends, kids to recreational areas in schools. It links sick to the hospital. It's vital as it links the overall economy. And when a blockage happens in these arteries of society, whether it's big, such as the blockage in Suez Canal, which I'm sure you have seen or heard of it in the last weeks, or as small as the traffic jams that you experience every day in your life, it stalls and affects our life routines. Now, consider when you're driving from Clemson University either towards Atlanta or Charlotte on Interstate 85. Remember the frustration you feel when you're reminded of or face the traffic in Atlanta. And some people might even suggest, why don't they add an extra lane? And when they do redevelop the highway, for example, in I-85 towards Charlotte, remember the frustration of facing construction that's been happening for a while now. And you must be complaining every time you drive that way and say, why can they expedite it? What if I told you that the construction processes or maintenance that makes you frustrated every day when you drive is a very small part of an extensive and complex process that you or the general public may have never heard of, which starts from identification of a transportation need in your local communities until it's being built. This complex and extensive process to take a transportation need from concept through construction is called project development process. And this process timeline is roughly 3 to 5 times more than the construction process because it has several phases. It involves various agencies from federal, state, and local. and considers various factors such as engineering, economy, environmental impacts, utilities such as water, internet, gas, and electricity, and so on. The main purpose of my research was to streamline this process and expedite it. And when I say streamline, I mean in terms of transportation project time, cost, and quality. But the question was how? How can I streamline this process? Now, according to the literature, one of the ways to streamline the PDP or the project development process is to identify best practices. Okay, it looks easy, but two main questions arises from here. First, what is the best practice? And second, how do we identify these best practices? These two questions guided me to the selection of an appropriate research methodology so I can achieve my research goals. Project development best practice is basically a method. It's a strategy. And it has two main criteria. One, that it consistently shows results superior to other methods. And second, it's used as a benchmark. Now we know what a best practices is, but the main question is how do we identify them? In order to identify PDP best practices, the research methodology I developed entailed 4 phases and 10 tasks. In research terminology, it's called mixed method sequential design. Basically, first phase was to understand the whole process in detail. Who is involved, what tasks are part of this process. And this was done by mapping the whole process by interviewing subject matter experts and analyzing documentation from all State Department of Transportation across the United States. Second phase was to involve all State Department of Transportation across the US and explore their project development timeline and their practices. The logic in phase 2 was that by identifying the timeline, I can group these states into 2 categories of top performers and poor performers. And then by comparing these 2 groups' practices based on their development timeline, I can identify the practices that can streamline the development process. But the problem was the second phase only provided the practices. It didn't tell me how these practices streamline the process. That's why in phase 3, by interviewing a small sample of these State Department of Transportation, I probed deeper to understand how these best practices streamline and expedite the process. And finally, in phase 4, by analyzing more than hundreds of documents, qualitatively and quantitatively, by conducting more than 100 hours of interviews, and by surveying more than 100 of subject matter experts from State Department of Transportation and their industry consultants. These best practices were identified, categorized, and recommended to Federal Highway Administration and the State Department of Transportation. My study's findings and result has provided an updated and current best practices that can streamline this process, and it increases the efficiency of project delivery. But the transportation industry is evolving in fast pace and it's changing continuously. So my study has also provided a model or the methodology for all State Department of Transportation across the US so they could systematically assess their current practices and improve their processes using this model. So next time when you are on the road and when you see a roadwork ahead sign, first, please slow down. It's really important. And second, do think about the time that has been spent on the development of that roadway that you're driving on, not the construction of it. Thank you.
All right. Thank you to Neen. Great talk. So, question from the Peanut Gallery. How do you think your project and model would change the effectiveness of project development?
So, well, it will change in terms of time. And once we change it in terms of time, once we streamline it, basically when I said it takes 3 to 5 times more than the construction process that you see when you drive in these roadways and everything, we can streamline this timeline to save a lot of money and also keep the quality same. So basically the 3 factors or the 3 metrics like time, cost, quality, if we affect one of these, it will affect indirectly the others as well. So that's how we can do it. And by implementing this model across the all state DOTs in the US, not only in one state. So bring consistency into that and kind of evaluate the situation, the current practices, because everything is changing and we have to keep updated this.
Awesome. Thank you, Taneen. Let's give one more round of applause for Taneen. And with that being said, we are going to move on to our next competitor, Lauren O'Connell. And in the meantime, we will give the judges another moment to wrap up their scores. And Lauren, if you want to go ahead and share your screen. All right. That brings us to our next contestant. Next up, we have Lauren O'Connell. Lauren is a PhD candidate under the advisement of Vincent Richards. She's in the Department of Biological Sciences under the College of Science. And today, she's going to be talking to us about friend or foe, the role of oil streptococcus in early childhood tooth decay.
Nailed it.
All right.
Testing, Lauren O'Connell.
All right. Thank you, Lauren. Whenever you are ready to begin, please feel free to do so, and good luck.
Thank you. So what if I told you that you could chew a piece of gum, suck on a mint, or even eat some ice cream, and that this would prevent you from ever having to go to the dentist to have a cavity filled? While this may sound too good to be true, the research that I will present today will outline a new method developed in my lab that will aid in the creation of an oral probiotic that can be used as a preventative measure and treatment method for dental cavities. The current methods that are used for identifying important microbes in dental health and disease can often only determine bacteria down to their genus level due to the similarity of their DNA sequences. This would be like somebody saying that they can identify an animal as a dog, but they don't know if it's a wolf, a coyote, or a domesticated dog. Because of this, our lab has developed a novel classification method using a new portion of bacterial DNA that can identify the oral streptococci down to the species level. Oral streps play important roles both in maintaining tooth health and in causing disease. So knowing which species promote tooth health is necessary for the development of a viable oral probiotic. Tooth decay is a problem that is experienced by all age groups, but can be inflated in children due to their higher sugar diet, their more frequent snacking, and less education on the importance of dental hygiene. In fact, tooth decay in children is one of the most common chronic early childhood diseases worldwide, with more than 50% of children suffering from a cavity by the age of 8. And this number can be as high as 70 to 90% in children that come from lower income families or from underdeveloped countries. If tooth decay is left untreated, it is painful, it can lead to tooth loss, it can lead to decreased sleep, decreased appetite, and decreased ability to concentrate at home or in school, all of which can have lasting impacts on a child's health and well-being. Though you can't see them, there are thousands of germs, including bacteria and fungi, that live in our mouths. The germs that cause dental cavities live in a film on our teeth and use our dietary sugars as an energy source. From this utilization of these sugars, they will produce acids as an end product, which are then released onto the tooth surface and can cause our enamel to wear away. If left untreated, the decay will progress into the deeper layer of the tooth known as the dentin layer. And if still untreated, The disease will eventually progress into the root of the tooth, causing an infection that will lead to tooth loss. The largest group of bacteria that inhabit the mouth are the oral streptococci, making up over 50% of the entire bacterial community in the oral cavity. The oral strep are important both in health and disease, with some strep species, such as Streptococcus sanguinus, believed to promote tooth health by taking up space on the tooth and preventing bad germs from setting up camp, while other strep species, like Streptococcus mutans, are believed to be the main cause of dental cavities because of their acid-producing capabilities and their ability to both survive and thrive under acidic conditions. Because of the importance of the different strep species in the mouth, the first goal of my research was to develop a cost-effective method that would allow for species-level identification of the oral streptococci and to validate that this newly designed method would outperform the more commonly used gene sequencing method. The second goal of my research was to use the newly designed gene region to examine the community of the oral streptococci and how this community changes as tooth decay progresses from health to disease. So in order to achieve these objectives, 84 samples were collected from 46 children after receiving informed consent from a parent or guardian. Each sample collected was placed into one of 6 categories that represented the different stages of disease progression, with stage 1 being a completely healthy tooth without cavities, and stage 6 being a tooth with a severe cavity that had progressed into the dentin tooth layer. After the plaque samples were collected, the bacterial DNA was extracted from the cells, and once the DNA was extracted, the samples were prepared for DNA sequencing. When the samples were ready, they were sequenced using a MySeq sequencer from the company Illumina. And then after sequencing was complete, the data was analyzed using different computer software. And from that, the taxonomic identification of the bacterial organisms that were present in the plaque samples was able to be determined. After looking at the data, I was then able to compare the results for species identification using the 2 different methods. The commonly used older method was able to successfully identify 2 different species of oral streptococci, while our newly developed method was able to successfully identify 13 different oral streptococcus species. This resulted in our new method being able to identify 11 more species than the more commonly used older method. So now that we knew that the new method was better at identifying the oral strep down to species level, we wanted to observe which species were present during the different stages of tooth decay. Interestingly, our data found that there was only 1 type of strep sanguinus that was associated with health, while there were 7 different types of strep sanguinus which were associated with disease. This was surprising, as many of the previous studies that have looked at strep sanguinus suggest that it's mostly found on healthy teeth. Next, when looking at the data for strep mutans, which is often considered the main culprit of causing severe dental cavities, we did find that there were some types that were strongly associated with progressed cavities, but we also found that there were 3 types that were only seen in early stage cavities. This raises the question that the current broad level classifications of health and disease associated bacteria might not be accurate, and that in fact, different organisms within the same species might be associated with health, while other organisms within that same species might be associated with disease. So this is really important to know when trying to develop an oral probiotic, as only those organisms that encourage tooth health should be targeted. Looking at our results, if we had arbitrarily chosen to add strep sanguinus to an oral probiotic because it is often considered an organism found in health, we might have inadvertently chosen a type that could have encouraged cavity development rather than one that encouraged tooth health. So now that we know that there are different types of the same species that may play different roles in cavity development and progression, a study that determines what the job of these organisms is in the community and how it relates to cavities is needed. The data from this study and future studies can be used for the development of an oral probiotic, such as gum or mint, or even an ice cream, that can be used as a preventative measure or one that can specifically treat dental cavities. Thank you very much for coming to my talk.
Thank you, Lauren. Great tab. So, question from the crowd. Are there takeaways we can use from this research or potential future research stemming from it to support children's oral health?
One of the biggest things I think from this research that we can take away is that the importance of teaching children about dental hygiene is the best preventative measure, because if they take care of their teeth because they know it's good, then that will prevent them most likely from developing a cavity later on down the line.
All right, great. Thank you, Lauren. Great job. So we are going to give our judges a few more moments to wrap up their scores. And then next up, we have Bob Wen. So Bob, if you want to go ahead and share your screen. And one thing I want to mention, when contestants, when you are going, when we ask the question, if you could please leave your screen shared for the time being, it makes the broadcast look a little bit better. So thank you so much. Okay, some people in the chat have asked, how can we vote for the presenter with the people's choice? We will be displaying that phone number at the conclusion of the presentation to ensure that everyone has the same amount of time to be texted in and voted for. So that will happen at the end, which will be around about 12:15. And so on to our next competitor. Next up, we have Bob Wen. Bob is a PhD candidate under the advisement of Dr. Curtis Simon in the Department of Economics under the College of Business. And today, Bob's going to be talking to us about the power of marriage, causal effect of stable marriage on child's adult earnings.
Hi, Bob Wen, testing.
Great, Bob. Sounds awesome. Thank you so much. And with that being said, the floor is yours. So whenever you want to begin, please feel free to do so, and good luck.
Thank you. A stable marriage between parents plays an essential role in their child's well-being. Unfortunately, the percentage of two-parent families among all the families with at least one child has dropped over time in the United States, from 75% in 1968 to 64% in 2017. Have you ever wondered whether your childhood family structure or your parents' marital stability will affect your future earnings? I have observed that the child's adult earnings are associated with their parents' marital stability. The children who grew up in intact families where their parents remain married earn more than their counterparts reared by non-intact families for all ages. However, the correlation between them does not necessarily mean causality. My research is to determine whether there's a causal relationship between the parents' marital stability and the child's adult earnings. To identify the causal effect of a stable marriage on the child's adult earnings, I adopted 3 specification strategies. First, I included the parents' income and the parental educational attainment. along with the child's demographic factors in the model to disentangle the parent's marital stability effect from the other parental influence. You may ask whether the parental stability has the same effect on the female children as it has on the male children. I found that a stable marriage has a larger and more significant effect on the son's adult earnings than the daughter's. In other words, among children from intact families, sons earn more than daughters. Among the children from non-intact families, however, there's no such gender earnings gap. The parent's income and the parental education also matter. The marital stability effect on the child's marital earnings is increasing with the parent's income. That is to say, your parents' marital stability and your parents' income during your childhood reinforce each other in raising your adult earnings. I found a similar pattern for the better educated parents. A stable marriage between well-educated parents helps their children achieve a better life. The PSID I used have traced individuals in the United States since 1968. They followed the children as they grew older and formed their own families, making it possible to link the children's information to their parents. I drew a family tree to show this structure. The original family of Bob and May had two children, Amy and Grace. Grace formed her own family later and had children, Ivy and David. Bob and May remained married during Grace and Amy's childhood. Ivy and David were also raised by an intact family. By contrast, another couple, William and Emily, had two children, Jack and Ellen. Jack divorced during his daughter Lily's childhood. I would expect to find that, as adults, Lily would earn less on average than Ivy, David, and Alex. In my sample, there are more than 1,000 clans like these two. I assume that there's something in common that runs in the blood across generations of descendants. It is the clan-specific heterogeneity, such as family traditions and genetic characteristics that are identical for all the descendants within a clan. I fitted a clan fixed effect model to account for these unobserved clan-specific factors. I also use an instrument variable, the no-fault divorce law, for the endogenous marital stability to further alleviate the omitted variable bias. Using these strategies, I found that the children who grew up in families where the parents remain married earn around 10% more than their counterparts reared by separated, divorced, or other non-intact families after holding relevant factors constant and correcting the omitted variable bias. This effect goes through three channels. The parents with a stable marriage tend to invest more in their children's education and health. Their children are also more likely to have a stable marriage themselves. So, the education you receive, your health, and your marital behavior are the three mediator variables that represent the three channels through which your parents' marriage affects your adult earnings. Above are the major findings of my research. The positive and significant effect of a causal effect of a stable marriage on the child's adult earnings demonstrates the power of marriage. Thank you very much.
Thank you, Bob. Great talk. Okay, our question from the crowd. Why do you think the marital stability of parents influences sons more than daughters?
That's a great question. That's about the mechanism behind this effect. One possibility is that the parents will invest more in the son's education and health than daughters. So as a effect, the son's adult earnings would be higher than daughters. Thank you for the question.
Awesome. Thank you, Bob. Great job. Let's give one more round of applause for Bob. Okay, We will give the judges a few more minutes to wrap up their scores before we move on to our next competitor. And next up, we have Madeline McCarthy. Madeline, if you could please go ahead and share your screen.
Can you see my screen?
Yep, we are going to wait a couple more minutes until 10:46, so hold tight. So next up, we have Madeline McCarthy. Madeline is a PhD candidate under the advisement of Dr. Mark Burtwissel in the Department of Chemical Engineering in the College of Engineering, Computing, and Applied Sciences. And today, she's going to be talking to us about fluorescence multiplexing using spectral imaging and combinatorics.
Testing, Maddie.
Sounds great, Maddie. All right. Whenever you are ready to begin, the floor is yours, and good luck.
Cancer. This word alone is enough to scare most people. It is currently a leading cause of death in the world. A large reason for this is because cancer is incredibly difficult to treat. For this reason, Developing effective methods for cancer diagnosis and treatment is essential. The immune system is involved in almost every disease in one way or another. More specifically, it plays an essential regulatory role in cancer development and progression. As we can see in this figure below, a tumor microenvironment is comprised of the normal cells, molecules, and blood vessels that surround and feed the tumor cell. As a result, a tumor's response to a treatment is heavily influenced by the tumor's microenvironment. This influence results in the need for doctors to be able to characterize the composition of a patient's tumor microenvironment. This is done through immune profiling, which is the process where a mixture of different cell types is classified into the fractional composition of its components. The figure to the left shows some of the different cell types that are present in your blood. Immune profiling is determining the composition of these cells in your specific blood sample. This technique is used in research and clinical applications to better understand and diagnose disease states. Essentially, it creates a snapshot of a person's unique state of immune health. The typical approach to immune profiling has been using a handful of fluorescently labeled antibodies in a flow cytometry-based assay. In this process, antibodies are tagged with fluorophores, which are fluorescent molecules, to create fluorescently labeled antibodies. These antibodies bind to specific cell types, like the one shown on the previous slide, and then are analyzed using a flow cytometer, which is used to count cells and detect fluorescence. However, this method is limited to a small number of fluorescent antibodies per sample due to similarities between the fluorophores emission spectra, referred to as spectral overlap. Other methods have worked to overcome this, but they are expensive, difficult, and destroy the cells during data collection, preventing further analysis of the sample. So now imagine that these are 8 fluorescent dyes that you have on hand in your lab, and you want to use them to label different cell types. However, all of these dyes can't be used in a mixture together because of spectral overlap. For example, let's take a closer look at dyes 1 and 5. We can see that they are very close to one another on this spectrum. As a result, our flow cytometer isn't going to be able to differentiate between them. After going through the rest of the dyes and removing the ones that are too close to one another, we end up with 4 dyes. This is the major limitation to current methods. Now we're going to use these pictures of balloons to represent 2 different patients' tumors and imagine that the different balloons are different cell types. With a limited number of fluorescent antibodies, which were referred to as colors, these 2 patients look like they have the same tumor immune profiles and thus could have the same treatment plans. However, with a larger palette of colors, we can see that these 2 tumors are in fact very different, resulting in the potential need for completely different tumor treatment plans. We can use this example to show why having a larger number of available colors or fluorescent antibodies is essential for generating an effective treatment plan. My research, multiplexing using spectral imaging and combinatorics, or MUSIC, helps to fill this gap in technology. MUSIC probes are created from single and covalent combinations of existing fluorophores, as you can see in the figure below. This expands our palette of available colors, significantly increasing the number of analytes that can be measured simultaneously. My work here aims to attach MUSIC probes to antibodies in order to create a wider palette of fluorescent antibodies that can be used for more comprehensive immune profiling. Essentially, we are using combinations of fluorescent dyes to create new colors that can be used to detect immune cells. This method has been tested using a proof of concept experiment with a 3-probe experimental setup. For our purposes, we will refer to probe 1 as a single probe with fluorophore A, which is our red star. Probe 2 is a single probe with fluorophore B, which is our blue triangle. And probe 3 is a combination probe of fluorophores A and B, which is our green square. For this experiment, the probes are attached to antibodies and then bound to protein A beads. The beads are detected in a similar way to cells on a flow cytometer, which allows us to use them as a stepping stone before applying this method to human cells. For this assay, we created 3 different batches of beads, which you can see in the bottom left-hand corner. single positive beads, which are labeled with a single probe, double positive beads, which are labeled with 2 probes, and then our triple positive, which will have all 3 probes. Now looking at the right-hand side of the slide, we can see 2 heat maps. The top heat map shows the actual amount of each bead that we put into the mixture, while the bottom heat map shows what the flow cytometer predicted was in each mixture. On the y-axis, we have the different types of beads, and on the x-axis, we have the different mixtures that were loaded onto the flow cytometer. In the first column, we have a mixture of all 3 single positive beads, and we can see that we were able to detect equal compositions of each probe. In the second column, we again have a mixture of all 3 single beads, but this time there's twice the amount of probe 1 as the other 2, which the flow cytometer was again able to detect the fractional abundance or proportion of each. As you go down the columns, you can see the different mixtures of single probes and 2-way mixtures, double positives, double and single positives, and our triple positive. And as you can see, both heat maps look very similar, proving that combination probes can be accurately distinguished from the individual fluorophores that make up the combination when in a mixture together. Music can also be used for any application where understanding a patient's current immune health or response to a drug is relevant. Therefore, it can be applied to many other diseases and viruses, including COVID-19. My research aims to create a more comprehensive diagnostic tool to allow for more effective disease diagnosis and treatment. Thank you.
Thank you, Madeline. Great talk. So we have our question from the peanut gallery. Can you explain the importance of unmixing during the spectral cytometry results?
Yes, so unmixing is essentially just determining which of your different probes are in that mixture. So if you have a mixture of what I showed, 3 different probes, unmixing is just determining that those 3 probes are present in your sample individually.
All right, awesome. Thank you, Madeline. Let's give one more round of applause for Madeline. All right, we are going to move on to our next competitor, the final presenter of session 2, Pinar. If you could please go ahead and share your screen, and we will give our judges a few more moments to wrap up their scores and lead off with Pinar at 10:57. All right. We're going to go ahead and wrap up session 2 here with Pinar Oz-Mizrak. Pinar is a PhD candidate under the advisement of Dr. Jane DeLuca. She's in the Department of Healthcare Genetics in the College of Behavioral, Social, and Health Sciences. And today she's going to be talking to us about health outcomes of hematopoietic stem cell transplantation for infantile Krabbe disease.
Pinar Oz-Mizrak testing.
Great, thank you, Pinar. So whenever you are ready to begin, please feel free to do so. And as always, good luck.
Newborn screening for Krabbe disease would improve life expectancy and quality of life. This is the statement that I would like you to take away from my presentation. You'll be seeing this word in the presentation a lot. It looks like crab, but it is pronounced Krabbe after the Danish neurologist who discovered it over 100 years ago in 1916. So in that past century, have health outcomes for Krabbe disease improved? We'll talk about that. But first, what's newborn screening? You might recognize that blanket in the background. It's the standard issue hospital blanket given to every newborn. Chances are, if you've been swaddled in this blanket, you've also been through newborn screening. It's a procedure that pricks the heel of an infant to take 1 or 2 drops of blood, from which laboratories can test for over 70 different health conditions, which can benefit from early intervention. This early diagnosis and treatment saves lives. I'm going to be looking at Krabbe disease in regards to newborn screening. It's a genetic disease that's caused by a lack of this enzyme called galactosylceramidase. Now, without this essential enzyme, babies are born appearing healthy, but within the first year of life, they start to show symptoms like muscle stiffness, seizures, developmental decline. It's a very severe condition and it's fatal at around 2 years old on average. Krabbe disease is a rare disease. So what is a rare condition? It's considered something that's in less than 1 out of 10,000 people. Krabbe disease is in less than 1 out of 100,000, so it's extremely rare. But because it's genetic, it's inherited from parents who are carriers, but not ill themselves. And the incidence of being a carrier is only 1 in 150, so much higher than you might think. And from 2 carrier parents, there's a 1 in 4 chance of having an affected child. There's a treatment option for Krabbe disease through hematopoietic stem cell transplantation. I want to clarify that these are not embryonic stem cells. These are stem cells donated from adults, either through bone marrow or umbilical cord blood. As a note, if you donate your umbilical cord blood, you can save lives. Now, stem cell transplantation for Krabbe disease is controversial because there's been varying success. So I wanted to create a literature review looking at, is the stem cell transplant effective enough to justify newborn screening for this condition? Because we don't want to screen for a condition when we can't also provide an intervention along with diagnosis. So I performed a literature search looking at results of the transplant. And since it's so rare, there's only been 9 studies that include health outcomes of patients in the past 20 years, which is how long the stem cell transplant has been performed. I gathered data on 71 patients. Now, the results show that when patients are transplanted, affects their health outcomes. So although patients live on average for 2 years, 20% of them can survive to 5 years old without any treatment at all. Looking here, 40% of patients who received a transplant after they had begun to show symptoms of Krabbe disease survived to at least 5 years old. But the key finding is this one. 80% of patients who were transplanted before they had shown any symptoms of Krabbe disease lived to at least 5 years old. And this number is just at when the study ended, so they continued to live past that point as well. So we see that the stem cell transplant can delay disease progression, but it has to be performed before symptoms begin to show. So knowing that the stem cell transplant has to be done pre-symptomatically, just how early is that? And the answer is under 30 days of age. So even patients who were transplanted at a month and a half or 2 months old, although they might have had an increase in life expectancy, what was their quality of life like during that time? Patients who were transplanted later had delayed outcomes in their motor skills and language skills compared to patients who were transplanted under a month old who had better outcomes in communication, feeding, even being able to go to school. As a note, the first patient who was transplanted with Krabbe disease is now a teenager and has graduated high school last year. So now we know that the stem cell transplant is more effective than no treatment at all, but it's most effective when it's performed under 30 days of age. So how can we provide the stem cell transplant under 30 days of age to patients who aren't even showing any signs of this condition? Since it's such a rare disease, there's two ways. One is to know your family history, but that's only possible if someone in your family has already been affected by Krabbe disease and there is no treatment option for this family member. The only way to treat the firstborn member in a family with Krabbe disease would be to identify them through newborn screening and create them as candidates for the stem cell procedure if there is a donor match, which is why it's so important to donate your umbilical cord blood if you can. So that's why I came to this conclusion that newborn screening for Krabbe disease would improve life expectancy and quality of life. And as a note, currently in the US, Krabbe disease is screened for in a few states, but each state has to pass its own law to add a condition to its newborn screening panel. So recently, South Carolina did pass a law and will be screening for Krabbe disease in this state. Thank you.
Awesome. Thank you, Pinar. Great talk. So we have our question from the crowd. For Krabbe disease, when a child is past 30 days old, what has changed in the human body that makes it so difficult to use HCST treatment to its best capability?
Excellent question. So Krabbe disease is caused by that enzyme deficiency GALC, which is creating this toxic accumulation in the brain, and that's why those symptoms are occurring. Now, this accumulation is happening bit by bit, and at birth, it's not visible unless we do some sort of testing, brain scans, MRIs. So although it's not visible through symptoms, after 30 days old, within the body, there has been enough degeneration that this isn't reversible. Whereas before 30 days old is the best time frame to be able to provide the stem cell transplant so that not only can we delay disease progression, but degeneration hasn't occurred in the first place.
Awesome. Thank you, Pinar. Let's give Pinar one more round of applause before we wrap up our second session here. All right. With that being said, we are now going to go ahead and enter our second intermission. Pinar, if you could please stop sharing your screen. And we will see everybody at 11:17 for the beginning of our third session.
be alright. Give it up, gonna be alright. Give it up, gonna be alright. Alright, alright. With you, what do you got? Alright, alright. So slow. Give it up, gonna be alright. Alright, alright. With you, what do you got? Alright, alright. So slow. Give it up, gonna be alright. Give it up, gonna be alright. Give it up, gonna be alright. Alright, alright. With you, what do you got? Alright, alright. With love, give it up, gonna be alright. Alright, alright. With you, what do you got? Alright, alright. So slow. Give it up, gonna be alright.
Oh, I haven't got it yet. I got one a while ago and I called back and somebody.
Give it up, gonna be alright.
Allow me to introduce myself. Who knows what dastardly crime might perpetrate next? He's a very clever arch criminal who must be put away. Another innocent
Give
Shandy, if you want to please go ahead and share your screen. I apologize if I said your name wrong.
Can you see my screen?
Yep. So the little button next to the plus minus on the bottom right, that little presentation square, you're going to want to touch that. to put it in presentation mode.
Wait, is it this
Yep,
one?
right there.
Okay. Sorry, with the music, it's hard to hear.
Oh, that's okay. I hear you. Is it Shen Yi, by the way?
Yes.
Awesome. Okay. Welcome back, everyone. We are going to go ahead and kick off our third session here. To the judges out there that are still here and helping, again, thank you so much. If you could please now flip over to the third session link in the email that Eric Antonio sent you the other day for this session, that would be great. So our next contestant is Xinye Gao. Xinye is a PhD candidate under the advisement of Dr. Anna Hall in the Department of Teaching and Learning in the College of Education. And today, she's going to be talking to us about promoting dual language learners' social and emotional development through creative drama.
Jenny testing.
All right, thank you so much. Whenever you are ready to begin, please feel free to do so. And of course, good luck.
Thank you. Let's meet Lucas. This is Lucas, who is 5 years old. He's currently enrolled in Head Start, which is a federally funded early childhood program that serves children from low-income families. Both of his parents don't speak English, and he just started learning English at school. Lucas is a dual language learner. There are more and more children like him in the United States who speak another language at home and who are identified as dual language learners or DLLs. In 2017, approximately 23% of preschoolers were DLLs, and in Head Start, this number is even higher. In 2018, a third of children in Head Start were classified as DLLs. DLLs are a unique group of children, and they are often compared with their mono-English speaking peers in the classrooms. So research has indicated that being bilingual might have a wide range of advantages, including more advanced executive functions, better self-regulation, enhanced resilience. However, the reality is DLLs have consistently underperformed their mono-English speaking peers upon kindergarten entry and in many academic assessments. So many of them are also found to be at a higher risk of developing negative social outcomes because their English proficiency and stressors such as like poverty or discrimination. So there is a mismatch between the learning experiences DLLs need to meet their potential and the quality of experiences they're currently receiving in schools. And this is something that I want to be able to address. A few years ago, when I worked on the Theater for Youth project, I witnessed how drama can be used as a powerful tool to engage children and facilitate their learning. So I started thinking how I can integrate drama in early childhood program to help DLLs. So in the current study, I explored creative drama as one possible instructional approach to promote the social and emotional development of young children in Head Start. Here, creative drama refers to the dramatic experiences that design for the development of participants instead of preparing participants for a performance before an audience. So they focus on the process instead of the final product. 6 dual language learners enrolled in a Head Start participate in the study, and they're between the age of 4 and 5, and most of them speak Spanish as their first language. And our Lucas is one of the participants. So these children participated in a 9-week creative drama intervention, and they engage in small drama activities for 20 to 25 minutes per day, 4 days a week. And you may ask, what kinds of drama activities? So a few examples are movement activities, puppets play, role playing, or storytelling. And here is an overview of the research design. So both quantitative data and qualitative data were collected. In quantitative data, the scores from the social skills improvement system rating scale were collected before and after the intervention. And qualitative data, including classroom observations, intervention field notes, were collected before, during, and after the intervention. And results from quantitative data showed that participants showed a significantly improvement from pre-test to post-test in their social and emotional skills. And on average, participants scored about 6 points higher on the post-test. And 3 main themes were identified in the quantitative data. So participants demonstrated increased confidence in their social interactions. So they're making more eye contacts while interacting with their peers, and they're not afraid to speak up during classes. Under the second theme, displayed social interactions, 3 sub-themes were also found. So participants showed increased initiation of social interaction, improved cooperation skills, and also stronger awareness of forming social groups. The last theme that were found in the qualitative data is a better emotion management among participants. So the current study showed promising results and provide preliminary evidence that creative drama can improve DLL's social and emotional skills in the classroom setting. The study confirms previous studies' conclusion about the positive impact of creative drama and adds to the growing body of research on DLL-specific instructional practices in early childhood programs. And on the more practical side, we want to design classroom activities with the intention of integrating multiple intelligences and facilitating children's learning through different modes so that we can better serve the needs of DLLs and learn more from this unique population of children. And thank you.
All right, thank you, Shenyi. Great talk. So we have our question from the Peanut Gallery. Your research worked mainly with preschool children. Is this the optimum time to prevent the negative long-term outcomes that you described?
I think so. So because like preschool is the most important time for children to develop their social and emotional skills, that's when they started to have a lot of interactions with the peers, with teachers, in addition to just their parents when they're younger. So this is the time that the most influential time for young children. And preschool, they can have like bigger influence on their like kindergarten. That's when they started to taking those high stake tests. And then later also as they're like in elementary and even later when they're grown ups. So preschool is one of the most important time to promote their social and emotional development.
Awesome, thank you, Shenyi. How about one more round of applause? All right, next up, we have Stephen Vicchio. If you want to go ahead and share your screen, and we will go ahead and give the judges a few more minutes to wrap up their scores before we begin. So our next contestant is Stephen Vicchio. Stephen is a PhD candidate under the advisement of Dr. Rachel Getman in the Department of Chemical and Biomolecular Engineering in the College of Engineering, Computing, and Applied Sciences. And today, he's going to be talking to us about determining the nickel catalyst structure to aid in the development of new energy efficient materials. Can we get a mic check, Stephen?
Can you hear me now?
Yeah, we can hear you great. Thank you.
Sweet.
Okay, with that being said, when you are ready to begin, the floor is yours, and good luck.
Well, thank you for the introduction, Nick. As we begin today, I first want to emphasize that catalysts make the world go round. Wherever you may be this morning, you'll find that catalysts played a central role in the development of your environment. Catalysts are used in up to 90% of all chemical processes, which help generate the consumed goods throughout the world. Now, you can find catalysts in a variety of different places. For example, if you drove into campus this morning or were in a car, a gas-powered car, there were catalytic converters that helped convert CO into CO2. Likewise, you have catalysts that are in the plants around us. So chlorophyll acts as a photocatalysis that allows or that helps to generate the oxygen that we need to breathe here on Earth. So catalysts are really important. They do really make the world go round. And within my work, I'm specifically focused on a specific application of catalysis, which is associated with gas to liquid transformations. So the idea here is that we want to take a stranded natural gas at these remote shale gas sites and convert it into some high value chemical or liquid, or high value liquid in this case. Reason being is that it's often either wastefully flared or pumped back into the ground. And so it's much more economically viable if we could develop a catalyst to convert the low value gas into a high value liquid. Now, our research group is a part of the Inorganometallic Catalyst Design Center. It's called ICDC, and it's an energy frontier research center that is looking to address specific questions related to these gas to liquid transformations. And so within ICDC, we attempt to address fundamental questions related to the discovery of new catalytic materials. Now, you might think to yourself, well, what is a catalyst, Stephen? If we take this reaction diagram, which essentially shows the energy needed to go from your reactants to your product species here, you can imagine without a catalyst having this black line. Now, if we try and push a ball up this hill, the barrier that it must overcome is essentially what limits the reaction. So we'll need some amount of energy in order to move the ball over to the product side. However, when we focus in on catalysis with a catalyst, what we'll see is that that barrier is decreased. So again, if we try and move that ball over this hill, we'll see a much lower activation barrier. Now, what catalysts do is they decrease the activation barrier, which allows them to create less energy intensive reactions, which is really important as we move towards, as we try and make sure that we conserve as much energy as we can. And so what we often are considering when we do catalysis type research is asking questions specifically related to what is the catalyst structure and how does it function? And so my work will look at the first question, which is really what is the catalyst structure here? Now, in terms of the catalytic materials we're working with, we work with a structure called metal organic frameworks. So metal organic frameworks is essentially a combination of these inorganic nodes or metal clusters shown here with the orange circles, combined with these organic linker molecules shown here with these purple bars. And when we synthesize experimentally together, we can go ahead and create these porous crystalline three-dimensional structures shown here called metal organic frameworks. Now, these metal organic frameworks often look like Tinker Toys, and we can create different frameworks by changing out and using different inorganic nodes and organic linkers. Now, these metal organic frameworks are not catalytically active on their own. You often have to use some sort of experimental technique to actually embed the catalyst species, essentially anchor it to the inorganic node here. And so although MOFs have been shown to be catalytically active for these gas to liquid transformations, we often have questions about exactly what is that catalyst structure. And so as we look to design new catalytic materials, we really need to get a good idea of what the catalyst structure is, because that'll allow us to better understand how the catalyst functions and in turn help us design new and better catalysts. So my work is specifically looking at, again, the structure, and we focus on the structure here of a nickel-4 catalyst. So a catalyst containing 4 nickel atoms, and it's shown here on the right, both atomically represented up top, as well as on the bottom using just atom labels, so from the periodic table. And so it's the same structure here, And this is one of the proposed models. But what we see is that experimentally, we see that the nickel-4 catalyst structure is dependent on both temperature and pressure. So depending on what temperature and pressure you operate and this catalyst is exposed to, you'll see that the structure of the catalyst will change. And so this is particularly problematic because we need to have a good idea of what types of changes are undergoing. And in terms of experimentally, it's really, really hard to elucidate and reveal exactly what types of changes this catalyst is undergoing. So some of the questions that we can ask specifically within my work is, what is the most likely catalyst structure when exposed to hydrogen gas, as well as is water present on the catalyst? And so within the Getman Research Group, we use computer modeling to address these structural questions. So the methodology, we essentially took we generated a library of 300 different catalyst structures. And so you can imagine now each one of these colored Ys are a different catalyst structure that we created. We could take all of these structures, feed it into Clemson's Palmetto supercomputer, where we are now able to, for every single structure, calculate the stability of it at different temperatures and pressures. So for all 350 structures in the library, they were inputted into the supercomputer, and we evaluated their stability at those different temperatures and pressures, where we looked at temperatures ranging from 20 to 320 degrees Celsius, as well as pressures of hydrogen. Now, output from this model are these phase diagrams that reveal the catalyst structure under different sets of conditions. And so when we look at this figure on the right, each one of these colored regions represents a different catalyst structure. So it is if we were under, say, a set of conditions that the middle blue structure, that would be the structure we would expect. And so we essentially are able to garner information in terms of what the catalyst structure is. Now we're able to identify the different structures here. And so these are just, again, shown atomically and using the atom label representation. But my takeaway, or the takeaway I hope for this slide, is that you'll see that there's a variety of different structures that this catalyst can adopt under different sets of conditions. And so as we improve our understanding of the nickel-4 catalyst structure under different temperatures and pressures, we can further evaluate the nickel-4 catalyst function, and in turn help develop new catalytic materials. And so as we develop these new materials for the future, we can first understand what is the structure of the catalyst and how it functions. And specifically within my work, we investigate the nickel-4 catalyst structure under different sets of conditions in order to continue the development of nickel-based catalysts for those energy-efficient gas-to-liquid transformations. And so catalysts make the world go round. And with that, I thank you for your time.
All right. Thank you, Stephen. Great talk. So let's move to our question from the audience. From a cost perspective, will the cost to convert these low value gases into high value liquids be higher or lower than the value of your final product, which would be your high value liquid?
So that's a great question. I'll say it's going to kind of depend. So in terms of taking these low value gases, a lot of them are at these remote shale gas facilities, say in like North Dakota. And what they do is they either pay the carbon tax and they burn it, or they dump it back into the ground. But if we're able to generate catalysts that can function at these lower temperatures and more energy efficiently, we're able to economically create these higher value liquids. So I'll say it more depends on, there's more that needs to be addressed to really answer that question.
Great. Thank you, Stephen. Let's give him one more round of applause.
And thank you, Nick. It's been great today.
Yeah. Okay. So thank you again, Stephen. And we will now be allowing our judges another moment to wrap up their scores as we move on to our next competitor in session 3, Roshan. So Roshan, if you could please go ahead and share your screen.
know, this is, excuse me, a damn fine cup of coffee. Hot black coffee. Like this. It's the first. Like Christmas. Oh, man, that is the stuff. Nothing like a great cup of black coffee.
Okay. So next up, we have Roshan Venkata Krishnan. Roshan is a PhD candidate. He is under the advisement of Dr. Sabarish Babu in the Department of Human-Centered Computing in the College of Engineering, Computing, and Applied Sciences. And today, Roshan's going to be talking about how the presence and size of static peripheral blur affects cyber sickness in virtual reality.
Can you guys hear me?
Yep. Sounds great, Roshan. Okay. So whenever you are ready to begin, the floor is yours. And as always, good luck.
Virtual reality is all about simulating experiences. Using computer technology, it is now possible for us to both create experiences that resemble the real world we live in, or be completely different, thus allowing us to do things in the virtual world that we never can do in the real world. Unlike traditional user interfaces, VR places the user inside the interface. It is only logical that VR applications are currently being used in a multitude of settings, including medical therapy, industrial and military training, education, and entertainment, to name a few. Yet VR usage isn't as widespread like the smartphone, for instance. Why is this the case? Cyber sickness is and continues to remain the major hurdle to the widespread adoption of VR. This begs the question, what is cyber sickness? Often described as a feeling of discomfort by users experiencing virtual environments, cyber sickness produces symptoms similar to those of motion sickness, like nausea, dizziness, disorientation, sweating, and even vomit sometimes. It is usually associated with travel in these environments where users perceive their self-motion, also called vection, in the virtual world, yet remain relatively stationary in the real world. This sensory mismatch causes users to get really sick. Researchers generally use two common ways to address sickness, either by altering the way you move through the environment or by manipulating what users see. We were interested in looking at how static peripheral blurring ties into this idea of reducing cyber sickness by manipulating what the user sees. But wait, what is static peripheral blurring? Like foveated rendering, it is a technique where we choose to render the peripheral region in lower resolution and the foveal region in higher resolution, thus allowing us to reduce the cognitive or the load on the graphical processing unit of the computer. It normally requires an eye tracking component to determine where one's eyes are looking at, though this needn't necessarily be the case, seeing as how Oculus came out with their fixed foveated rendering technique in 2018. As such, researchers investigating this phenomenon focus more on optimizing the GPU performance and improving the user experience, more so than looking at them with cyber sickness in mind. We were hence interested in determining how the presence and size of these static peripheral blur windows affect the onset and severity of cyber sickness. Downstream of this, we also wanted to understand how users relate to users' spatial awareness and presence during the experience. For our static blurring technique, we implemented a Gaussian blur filter in the peripheral region and rendered the central image area in full resolution. We call this the central window. It's circular in shape and fixed at the center of the field of view. We placed users in a virtual 20 by 20 virtual maze. By carefully choosing textures and shaders, we increased both the realism associated with the scene and the amount of self-motion participants perceived. We designed a 3-session within subject study where the central window size served as the intervention. Each session was held on a separate day with at least a day's gap in between them. The 3 levels included a normal viewing condition with no blurring and 2 window sizes, small and large, that featured peripheral blurring. Note that window size corresponds to the size of the central window rendered in full resolution. On juxtaposing the levels of the intervention, notice how a small window corresponds to a larger peripheral blurred region and a large window corresponds to a smaller blurred region. We devised two tasks for the study, namely a search task and a spatial updating task. The search task was used to guide participants along a predefined path through the maze, for which 100 red balls were sequentially spawned one after the other along this path, each one appearing at a nearby location after the previous one had been collected. This way, participants were engaged as they traversed the trajectory of the maze. The spatial updating task, on the other hand, was used to measure participant spatial awareness. How do we do this? We used target objects like this bottle and presented them twice, the first time highlighted in yellow and the second time highlighted in blue. When participants encountered the object for the first time in yellow, they then collected it and followed the red balls until they collected the object or they encountered the object in blue, upon which they were asked to point to the location where they collected the first bottle. We used this to measure the accuracy of their spatial awareness. We recruited 30 participants for the study, half of which were female, and made sure that all our participants had corrected to 20/20, if not for normal vision. After obtaining participants' consent, they were asked to outline their previous experience with VR, along with standard demographic questions. What did the study entail? Participants were first briefed about the study, following which they were trained to use the Vive controllers to locomote within the scene. We then administered the first instance of a simulator sickness questionnaire to get a baseline measure of their sickness prior to the simulation. They were then seated on a fixed chair and were instructed to report their levels of discomfort whenever asked. Participants then began performing the spatial and search updating tasks in parallel. Here's a live rendering of what the experience looked like. Following this, participants took the second instance of a simulator sickness questionnaire and then proceeded to engage in semi-structured interviews after gathering their data and presence. In the semi-structured interviews, we probed into their perceptions across sessions. So what did we find? We found a significant effect of the intervention in predicting discomfort scores, where we observed that the sickness levels in both conditions with peripheral blurring was associated with lower levels of sickness. However, we found no significant differences between the large and small window conditions. So it seems to be the case that introducing static peripheral blurring reduces sickness. But why? It's because your perception of motion is stronger in the peripheral region than it is in your foveal region. With respect to spatial awareness, we found no significant differences between conditions. So by using our blurring technique, we were able to reduce cyber sickness without compromising spatial awareness. Why is this the case? Compared to field of view restrictions, where the peripheral region is blackened out, our technique preserves information. What did our interviews tell us? Participants commented that they noticed something off with the small window condition, but couldn't exactly articulate what this difference was. Hilaire explains that this presence is due to increased realism. We additionally think that lower levels of sickness is another reason for this preference. So in conclusion, we were able to show that static peripheral blurring reduces cyber sickness. More so, this technique doesn't come at the cost of user spatial awareness and presence. What does this all mean? Static peripheral blurring is a promising technique. Why? Because it mitigates cyber sickness without compromising other aspects of the experience. It lowers the load on the graphical processing unit. And now we don't have to rely on resource-intensive gaze contingent eye tracking in order to achieve these goals. With that, I would like to thank everyone for listening, and I'd like to take questions.
Thank you, Roshan. Great talk. Okay, let's get our question from the audience here. Would some games or other applications be hindered by the peripheral blurring that you described?
Yeah. Could you repeat that question? I heard part of it.
Would some games or other applications be hindered by the peripheral blurring?
Yeah, actually, games that involve a lot of rotational movements induce a lot of cyber sickness because rotation causes you to perceive different angles of motion as well. So games that have a lot of rotation causes users to get more sick. And research has shown that blurring the field of view during these rotational movements can reduce sickness. So games that involve a lot of turning around and ducking and going up and down, that involve changing your posture, causes users or causes people to get more cyber sick.
Great, thank you, Roshan. So with that being said, we will now move on to our, one more round of applause for Roshan, I apologize. The crowd's wild today, we gotta hold them back.
This thing is crazy.
Okay. So We will now move on to our next competitor, Michelle, and we will give the judges a few more moments to wrap up their scores, and we will begin in a couple minutes. So next up, our next contestant is Michelle Eichinger. Michelle is a PhD student under the advisement of Dr. James Spencer in the Department of Planning, Design, and the Built Environment in the College of Arts, Architecture, and Humanities. And today, she's going to be talking to us about spatial mismatch, food insecurity, and the food retail environment accepting SNAP, food stamps.
Can you hear me? Michelle Eichinger testing.
We can hear you great, Michelle. Thank you.
Okay.
All right. Whenever you are ready to begin, the floor is yours. And of course, good luck.
Thank you. Food insecurity, or the lack of adequate food to live a healthy life, is a chronic issue in this country. Nearly 1 in 8 Americans are food insecure, which is about 40 million American adults. Food insecurity leads to very poor health outcomes. And in this country, we have this paradox where it's more of the quality of the food as opposed to the quantity of food that leads to these chronic diseases, especially obesity. It's energy-dense foods that are more affordable, which then leads to poor health outcomes, such as type 2 diabetes, malnutrition, and obesity. In this country, we have the Federal Supplemental Nutrition Assistance Program, also known as SNAP, also known as food stamps, or in the stores, you might see this little sign that says EBT. This is the financial assistance program to help increase the purchasing power of income-eligible households to purchase foods that at SNAP authorized vendors in the country. And they use cards that operate similar to a debit card or EBT card, and that's how they purchase these foods. So despite having the largest anti-hunger program in this country, we still have this food insecurity problem that leads to social and health issues. But most of the research has been focusing more on the consumer perspective and less on the retailer's perspective. And SNAP authorized retailers are administered at the federal level, unlike the consumers, which are administered at the state and local level. So there's a disconnect on what the food retailer types that accept SNAP and where they're located and the type of food that they provide and the SNAP consumer needs. And so in this study, I looked at the SNAP retail environment, mainly because food systems is not adequately addressed in urban planning, and more specifically, food planning as it relates to food insecurity. So in this study, I looked at an innovative approach called the Jacquard Index, which is typically common in artificial intelligence. And what I did was take the kernel density of SNAP retailers and create a raster map and use that almost as an image and look at the spatial similarities and spatial comparisons of the food environment, looking at healthy versus unhealthy SNAP retailers at different distances at the local level. And I also then weighted that with SNAP participation at the local level. So how did this work? So the USDA, United States Department of Agriculture, administers the SNAP program, and I downloaded all the SNAP retailers in the country, at least within the 48 states in the District of Columbia, and geocoded those across the entire country. I then categorized them as healthy or unhealthy, which is based on the Centers for Disease Control criteria on identifying the food retail environment type. Then I used the CDC's 500 Cities program to look at obesity and diabetes and SNAP participation at the local level, at the census tract level. And I wanted to have at least one city in each state. In this study for GRADS similarity, I focused on the city of Atlanta. And the SNAP participation data that I gathered is actually from the community survey or the census data. So in terms of weighted and unweighted, I looked at first the concentration of healthy food retailers, healthy SNAP retailers, which are your supermarkets and your farmers markets, produce stands, and look at the relationship where they're located with the unhealthy food retailers, which are your convenience stores and your corner stores, and look at the concentration of them. And so in the unweighted study, so this is strictly looking at the spatial differences of the healthy and the unhealthy retail environment, I found that at the half-mile neighborhood distance, we see the biggest mismatch. And that's significant based on the table of significant values of the GRADS similarity index. What is more interesting about this is that when we look at food insecurity, we are looking at food access and its relationship of how do we get there. So at the half-mile level, people are more likely to walk. And so in these lower income areas and urban areas, they're less likely to have a vehicle. And so they're more likely to walk. And the 1 mile radius is the typical standard of food access that is USDA defined. So we see that there is a definite mismatch of concentration of SNAP retailers from healthy and unhealthy. And then when you factor in the participation of SNAP, people who participate in SNAP, so I weighted each SNAP retailer based on the population rate of SNAP participation. And this was further identified as a mismatch. So basically what we're seeing here is that people who are more likely to participate in SNAP have higher, there's a higher concentration of unhealthy SNAP retailers in the half-mile neighborhood as opposed to the healthy SNAP retailers. So this reinforces this behavior of unhealthy eating and affordability when the retail environment is more attracting to low-income areas that are selling unhealthy foods. So what does this actually mean? So what we are seeing from the public health perspective is that, yes, the food retail environment could definitely have a relationship with health status. So we see poorer health outcomes, obesity and diabetes, and higher SNAP participation in areas that have more unhealthy SNAP retailers compared to the healthy SNAP retail environment. This is further exacerbated by people who participate in SNAP. So there is a mismatch in the SNAP retail environment. And what this does is it informs planners and decision makers on where to target strategies to improve healthy, affordable foods in these vulnerable neighborhoods. And then look at local planning can work with these small retailers to meet the criteria to be a SNAP vendor and provide the infrastructure needed to help sell healthy foods. And then with that, I want to take the time to thank everybody, and I'll answer any questions.
Great. Thank you, Michelle. Great talk. All right. We got our question from the audience here. Why do you think the concentration of unhealthy SNAP retailers was significant on the neighborhood scale more than others?
Well, we're looking at the local scale where people actually can access and walk to food retailers or just retailers in general. And why do I think that? Well, it has to do with a couple things. Healthy foods tend to be more expensive, and you need to have, and also more perishable, and you need the retail space, the floor infrastructure and the refrigeration units, and you need the infrastructure and the floor design to do that. And oftentimes in these urban areas, you may not necessarily have that luxury of land to be able to have the infrastructure needed to sell more healthy foods. So that could be one of the issues. Now, again, one way to address that is working directly with these small retailers to provide alternative ways to sell healthy food without having to expand that land or expand their stores.
All right. Thank you, Michelle. Let's give her one more round of applause before we move on to our next contestant. Okay, so we will give the judges a few moments to wrap up their scores before we move on to our last contestant of session 3 and the last contestant of this competition. Bridget, if you want to go ahead and share your screen, and we will begin in a couple minutes. We're on the ones and twos, just spinning the smooth grooves, keeping the ambience up. Hey, Bridget, is that black box on your screen?
Can you hear me?
Yeah. You're fine.
Black box?
Yeah, never mind. Something on the screen went away. Okay. So, our last contestant of the day, we have up Bridget Blood. Bridget is a PhD student under the advisement of Dr. Jeff Wong in the Department of Forestry and Environmental Conservation in the College of Agriculture, Forestry, and Life Sciences. And today she's going to be talking to us about functional morphology of shortleaf pine, Pinus encinana fire-adapted traits.
All right, so y'all can hear me, right?
Yeah, sounds great. Okay.
Awesome.
Well, whenever you are ready, Bridget, the floor is yours, and good luck.
Thank you. Well, howdy, folks, and thanks for coming to my talk. Shortleaf pine is one of the most important conifers in the southeastern United States for lumber, plywood, and pulpwood. Prior to European colonization, shortleaf pine dominant forests covered roughly 70 to 80 million acres. Today, there are only about 6.1 million acres remaining, suggesting a decline of greater than 90% due to increased suppression of wildfires and reduced prescribed burning. Shortleaf pine is a fire-adapted species that requires frequent surface fire to maintain its dominance in forest communities. Wildfire is a natural ecological process and one of the most common disturbances in many forests. It can be detrimental to seedlings and saplings, often leading to the death of the stem, also called top kill. Frequent fire occurrence has led to the development of 2 fire-adapted traits in shortleaf pine: resprouting from the basal crook after top kill and developing thick bark to avoid top kill. The basal crook, a part of the stem that grows horizontally underground before redirecting to vertical growth, has dormant buds insulated from fire by a layer of soil and duff. With the development of that crook, shortleaf pine can vigorously resprout after top kill, which gives shortleaf pine a regeneration advantage over other less fire-adapted tree species. Wildfire plays a key role in encouraging shortleaf pine natural regeneration and later survival. Repeated top kill often prevents that pine tree from growing into the forest canopy. To be recruited into the forest canopy, shortleaf pine must avoid top kill by eventually growing its bark thick enough. That thick bark protects the tree from cell death, which occurs as the inner layers are heated to temperatures above 60 degrees Celsius, leading to the eventual death of the tree. Depending on fire characteristics, a bark thickness of 5 to 18 millimeters is necessary to insulate the cambium from temperatures above 60 degrees. Both adaptations are beneficial to shortleaf at different life stages. As shortleaf pine grows in diameter, the basal crook gets swallowed by the tree stem, likely decreasing the tree's ability to resprout. But the bark is also growing thicker, likely increasing the tree's ability to survive fire. So where is that sweet spot in diameter to incorporate prescribed fire into the landscape? Shortleaf pine is one of the most valuable pines in the Southeast US. Thus, knowing how to properly manage fire in current shortleaf pine communities, shown in red on the map, is essential for restoring shortleaf pine forest throughout its historic range, outlined in black. The use of prescribed fire too soon, in other words, before that basal crook is fully developed, could result in the outright mortality of shortleaf pine seedlings, whereas a high-intensity prescribed fire could also kill many shortleaf saplings during the period of shifting fire adaptation strategy. Therefore, a clear understanding of these critical life history events is necessary for the proper management of shortleaf pine. This research examines the shift in fire-adapted strategies for shortleaf pine across its life stages, from seedling to mature tree. The main objective of our research is to characterize shortleaf pine resprouting after top kill and document the growth of bark in relation to stem size and age. We hope to better understand when that shift in fire defense strategy occurs so that we can incorporate our findings into shortleaf pine management plans. To do this, we top killed 158 shortleaf pines in the Ouachita National Forest in Arkansas in the summer of 2018. We measured diameter, height, bark thickness, and marked the location of each stem. So, one growing season later, we determined whether a stump was live or dead based on the presence or absence of sprouts. We developed a logistic regression model with a log-it link function to predict probability of survival, live or dead status, for a given stump as a function of diameter. We also created a linear regression with diameter, age, and the interaction as predictors for bark thickness. Excuse me. We found that larger stumps are less likely to resprout or survive top kill, which we expected based on that growing trunk swallowing that basal crook. This figure shows the probability of sprouting from a stump by diameter. The horizontal axis is diameter in centimeters, whereas the vertical axis is status, live dead, with live along the top of the figure. We considered a tree more likely to die from top kill if the probability of death was greater than 50%, as shown by the horizontal dashed line. Right of the vertical dashed line in pink shows the diameter when a stump is more likely to die after top kill, right around 15 centimeters. Oh, sorry, excuse me, right around 12 centimeters. So this figure shows that smaller trees are more likely to sprout after top kill. Pretty simple. We also found that diameter, age, and the interaction effect of all significant predictors are all significant predictors for bark thickness. Diameter in centimeters is along the horizontal axis, whereas the vertical axis shows bark thickness in millimeters. Age is shown in these beautiful Clemson colors, with the smaller purple circles indicating younger ages and the larger orange circles. The horizontal dash line shows the minimum bark thickness necessary to insulate those inner tissue layers during the fire that I mentioned earlier. To the right of that vertical dash line in green shows the diameter when a stump is more likely to survive fire, about 6 centimeters or 7 years old. So this figure shows that large trees are more likely to avoid top kill from a fire. We found that the defense mechanism shift depends on tree size. Smaller trees sprouted more readily after top kill, while larger trees were less likely to survive top kill. We hypothesized that the larger trees did not sprout as readily because the thicker bark would have protected the inner tissue layers, resulting in a decrease in the probability of mortality. This indicates a shift in fire defense mechanisms throughout the life stages of the shortleaf pine. As a forester, my research is useful when it impacts the people managing the land, the boots on the ground. So because prescribed fire is a common management tool, our research will help to establish burn intervals for shortleaf pine communities to avoid unwanted seedling and sapling mortality. Incorporating other aspects of our research will lead to determining the ideal fire return interval, whether for seedling recruitment, when you would burn earlier, or for state establishment, about 7 years. Thanks everyone for coming to my talk, and I'm excited for the questions. Sorry, I'm getting over a cold right now.
Oh, you're okay, Bridget. Great talk. All right. Question from the peanut gallery. You mentioned in your results that tree age is not a significant predictor of stump survival after top kill. Does that suggest a lack of correlation between age and diameter?
So not entirely. So for this, when we were looking at tree age, those, oh no, let's go to this picture. As that stump grows, the older trees are more likely to have larger stumps, but also if a tree is repeatedly top killed, the age would not be shown in the stump. So looking at the age of a tree that say maybe was top killed 3 or 4 times could be 15, 20 years old. And therefore it's not super linked to the survival. Generally speaking, when you're working with above ground trees, diameter and age of that top portion is closely related, but not so much for the stump age relation.
All right. Thank you, Bridget. Great talk. Let's give Bridget
Thanks.
one more round of applause as we round out our last session. All right. Thank you, everyone. I think everything's going really well so far. And so now that we have wrapped up all of our contestants today, and so now is the time that I will bring out the People's Choice. There is a phone number that I am going to share on my screen in a second here. And what you will do, second here. Okay, so we now have the opportunity for the audience members to go ahead and vote for their favorite contestant today, a brand new thing to GSG competitions. And so what you will do is text finals in the number of your favorite presenter to 833-344-0159. We're going to leave the voting open until about 12:30. And when you text the number, it should give you a confirmation that you voted. And just a heads up that you only get one vote. It will not allow you to vote more than once. So with that being said, we are going to tally up our scores and see you in about 20 minutes to announce our winners for the award ceremony. everyone, I'm going to jump on real quick. I just want to let you know that we are still in the process of tallying up the scores. So we are going to delay the award ceremony until 12:40. In the meantime, the People's Choice Award is going to close in the next minute at 12:30. Again, we will be back with you at 12:40. Thank you for being patient with us. All right, everyone. Welcome back. We thank you for being patient. Some of the scores were really close, so we wanted to make sure that we had everything correct before we went ahead and started the award ceremony. I just want to take a second again to thank everyone that was part of making this happen. This was a big operation, and I'm very proud of how well today has went. I think it could have went just about as well as we could have asked for. And I think all the presentations were phenomenal. And we really did a great job of showcasing some of the great research that's done in all of the colleges here at Clemson University. And one thing I wanted to mention is that we have also added a digital badge this year. So Grad 360 partnered with Clemson Online to make digital badges for each of the winners. The badges reflect the transferable skills and competencies demonstrated throughout the competition today. It is a wonderful way to promote your research and how you've been able to communicate it effectively. And these will be displayable on your LinkedIn profile and other platforms out there. So with that being said, I want to go ahead and hand things off to Dr. Tia Damas, who is going to say a few words before we kick off this award ceremony.
Hi, Nick, thank you so much. Can you all hear me okay? Thumbs up? Okay, fantastic. Yeah, thank you. So good afternoon, everybody. The time has arrived for the presentation of awards. So my comments, my statements will be rather brief. I realize that I stand between graduate students and lunch/cash prizes. So not a good idea. Let me just launch into this. So I want to echo Dean Lope's opening remarks and extend my gratitude to each of this year's competitors as well as their faculty mentors. You invited us into your studios, your labs, your fields, your classrooms, and other research spaces, and engaged us in interdisciplinary knowledge exchange. Each of you inspired to make the complex accessible, and you are actively disseminating your research in a range of scholarly, professional, and public outlets. For most, if not all of you, this is only the beginning of what will undoubtedly be inspiring and impactful careers that advance academia, government, nonprofit, industry, and entrepreneurial ventures. To all of the organizers of this wonderful event, thank you for creating conditions in which Clemson's master's and doctoral students are challenged, supported, and celebrated. Your commitment to one another and to the graduate student community is admirable. I also want to thank the Grad 360 faculty mentors who participated in both the design and some of the execution, the dress rehearsals, as well as the Grad 360 team in the graduate school. A shameless plug, please check out the graduate school's website, our social media platforms, including Twitter. Check out the Clemson's Grad 360 website. That's clemsongrad360.com. There's lots of information, events, news, announcements, and of course, this event will probably be promoted on all of those channels. So check out all of that. I would be remiss if I did not share that I'm also personally proud to witness and support Nick Gregorich and his leadership development. Nick, you inspire me to bring my whole self to my mentoring relationships. I'm humbled by your drive for excellence, and I'm honored to continue to work with you and GSG this next year. And yes, while GRADS 2021 is a competition and awards will be made, when all of you communicate and disseminate your research and discovery with diverse audiences, we all win. Congratulations to all of you. We are extremely proud of your accomplishments and successes.
Thank you, Dr. Damas. That was really nice. So we have one more speaker before we jump into the award ceremony. Our Vice President of Graduate Student Government, John Porter, is going to say a few things. So John, the floor is yours.
Hello, everyone. Great to see you all again. Just some short commentary from me. I think Dean Lopes, Dr. Dumas, and Nick Gregorich has said pretty much all of it already. Just want to thank you all for coming out. always great to see student-led research events by students for students. It's always a pleasure to find out all the research that's going on. I know for a fact I probably wouldn't have a chance to know everything that's going on campus if not for events like these. So these events allow people both on campus and off campus and around the world that are in Clemson's family to get together, share research, learn from each other, build each other, and critique each other, because that's a part of what we do, and we're not ashamed of it. We think it makes us better researchers, better people, and better presenters. So without further ado, I'm gonna let you get to the awards. Before I go, I would like to say it's been a pleasure serving as your elected official vice president for graduate student government. I will be leaving you all. I've successfully defended my dissertation on Monday. So I'm now Dr. John Porter III. Thank you all. Thank you. So I'll be heading out, but I'm very, very confident that I'm leaving you in great hands with Robert O'Hara and Abby Roberts, your new GSG president and vice president. They'll be taking over the term starting next Thursday, and I'm sure great things are to come into the following year. So for all of you that are still out there, keep pushing. I know you'll make it with the support of friends and family, all of which we're also here on Twitch and Zoom. So I want to thank you all for coming to support your family and friends, students. And it's been a pleasure, and hope to see you all in the future walking across that same stage right behind me. Make sure that you build up this event from the years to come, because just like we're doing it for the students here, we're going to need some of you that are enjoying these events to come join us at GSG so that you can help us build it for the people coming behind you, because that's how we all build and continue to mentor. Thank you.
Thank you, John, and congrats on your defense. What a great accomplishment. Okay, with that being said, let's go ahead and get this thing rolling. So first up, our third place contestant, who will be winning $250 in our first ever virtual grads competition. And without further ado, join me in congratulating Sarah Kravasik for placing third. So one thing I forgot to mention, when we bring everybody up, we are going to take a screenshot. So I'm going to count down and go ahead and smile or wave or whatever you want to do, but we want this for our own records. So in 3, 2, 1. All right. Again, congratulations, Sarah. Let's give her one more round of applause. Okay. So next up, our runner-up, who will be winning $500 today. So yeah, our second place winner. Please join me in congratulating Bridget Blood. Congratulations, Bridget. Congrats, Bridget. I know you emailed me at 2 in that you weren't feeling well, so you should be very proud of yourself that you were able to pull this off. Very proud of you. So I'm going to count down in 3, 2, 1, and we're going to go ahead and take another picture. 3, 2, 1. If you want to say something, by the way, go ahead. You don't have to hide behind being muted.
You're talking to me, sir. You totally froze.
Yeah. Oh, it's okay.
Oh,
right.
Thanks.
Yeah, congratulations.
Thank you.
I really enjoyed your presentation. Okay. So last but not least, our first place champion, the GRADS champion, the person that is winning a grand with GRADS. What an accomplishment. I think you should be really proud of yourself. There were a lot of great presentations in both the preliminary and final phases of this competition. The scores were really, really close. So without further ado, congratulations to Lauren O'Connell from the College of Science. Lauren, how are you feeling? Congratulations.
Very surprised. Thank you.
Congratulations. All right, so I'm going to count down in 3, 2, 1, and we're going to go ahead and take your picture. 3, 2, 1. Okay. Again, congratulations. What an accomplishment. I think you should be really proud of yourself, and what a great way to end the semester. So, all right. So now our new category this year, the People's Choice, who will be winning $100. Thanks to everyone who voted. I've seen the final scores, and I think we had over 100 people, or almost 100 people vote. So a lot of activity, and I think that's really awesome. So without further ado, the People's Choice winner for your 2021 GRADS competition is Paritra Mundell. Congratulations, Paritra.
Yay! Thank you.
Congratulations, Paritra. So let's go ahead and take a picture. We're going to go 3, 2, 1. All right. Wow. I guess that's it. This has been quite the ride, and I'm very proud of you all. This went again about as well as it could have. Congrats to all the winners. I will be reaching out to you in the coming days about getting your award to you. And with that being said, I guess it's time to sign off. It's been a blast being your host for this for the last, 3.5, 4 hours. I am exhausted, and it's been a lot of fun. And we will see you guys in the fall for 3MT, and hopefully we are back in person again. So with that, I'm going to sign off, and everyone have a great weekend. It's been a blast.